That you can hear. Sorry for this delay. We're about almost 12 minutes late. We're trying to come up with this time in the coming presentations. So my name is Mohamed Al-Gharous. I am delighted to chair this session with my colleague Krishna Dikova, who is a reporter for this session. We are very tight on time, and it happens that today is my birthday, so I'm happy to do this.
Congratulations.
So without delay, I will give the floor to our colleague José Luis Arguez Ortica, University of Barcelona, who is a keynote speaker. He's going to talk to us about affordable high through-output phenotypic techniques to assess crop resilience to salinity and water stress. So, Jose, I will give you the floor.
Okay.
In fact, one moment, we have real tight in time. I hope that you have 30 minutes, but if you could save us some time, it would be good.
Probably no, but if you let me know in advance, 5 minutes in advance, I will just to cut and finish.
Okay, thank you.
That should be the easiest.
Thank you. The floor is yours.
Yeah. Okay, thank you, Chairman. I'm sorry for the delay. So my experience is mostly...
Can you put the presentation on?
Still seeing the screen here, but can...
Okay.
No, okay. Yeah, it should be. Okay, so let's... how to advance here is frozen. No. Sorry, no. Now it's probably working. It's frozen.
No, this one is not.
No, yeah, it's not
just
everything.
to see what's going on in it.
Yeah.
He's going to give you the hand on that.
Well, in the meanwhile, yeah. Okay, so, yes. Full screen, yeah, but full screen. No, yeah, I asked him. Yeah, okay. So the presentation, as you see, will be the ways we may have to phenotype in the wide sense, not only phenotype for breeding purposes, but also phenotype for crop monitoring, and especially with the idea of low cost and projects. Next one, please, because it's not working. Okay, thank you. As you may see, and yesterday was clearly stressed, the Mediterranean and Middle East area are the hot spot in terms of salinity. And one of the topics concerning salinity, what particularly I'm most interested in salinity under irrigated conditions, which are actually the lands. The irrigated areas are the most important, and those are really exposed to salinity. Thank you. Next one, please. Yeah, so the line of the presentation will be what tries to phenotype coming from a physiological understanding. So if we know the physiological response of the plants to salinity, we may try to think about what are the traits crucial. The second, and within this, I will talk a little bit on tools to phenotype growth and senescence, to phenotype gas exchange, transpiration and photosynthesis, and to phenotype ion balance and also nutrient metabolism, which I understand are also important. And finally, I will stress on how to phenotype, especially with the idea of low-cost approaches, platforms and tools. And the last point is that there is not only remote sensing, but there are also other phenotyping techniques that may be interesting or important in some cases. So I use a presentation of Anna Moons and Mark Tester, one of the most cited reviews on salinity. I will say the most, because it's very pedagogic in the sense that salinity, the response of plant to salinity is a two-step response. The first one is an osmotic stress, which means a rapid response similar somehow to water stress. And the second on time is ionic stress, which means it's a longer term and eventually may produce an accelerate senescence of the plant. And this is why there are two factors. The osmotic stress is decreasing growth. So if we are able to phenotype growth, we will be able to catch the response of the plant. And on the long term, is also being responsible of senescence. So if we are able to catch senescence, there is also another interesting trait, especially for long-term response to salinity. So the point is how we may monitoring senescence and plant senescence in one hand and growth of the plant on the other. There are other studies that also put a lot of emphasis in water stress and salinity affecting gas exchange, basically photosynthesis and transpiration. And this is why also there has been a lot of work devoted on phenotyping those characteristics. The point is that when we consider water stress, ionic stress as a first inducer of that response, the leaf expansion is by far the first affected. So even if the salinity is mild and is not apparently affecting the photosynthesis, what will affect by sure is the leaf expansion. So if we are able to catch differences, if the plant is growing faster or not as happy, we may catch somehow the mild salinity, while photosynthesis usually is affected when the ionic stress, when the water stress, when the leaf potential is more negative. Okay, so this is one point to consider. And the osmotic stress limits especially the capture of the photosynthetic radiation, more than the radiation use efficiency, more than photosynthesis at the leaf level in many cases, okay? The other point is that the stress, only when the stress is severe, the accumulation of solutes, or for example, abscisic acid that is causing stomatal closure will take place. So again, salinity, severe salinity or long-term salinity will produce stomatal closure and decreasing photosynthesis. But on the short term, plant growth is the most important. But somehow those characteristics, plant phenotyping, transpiration, photosynthesis is also important in top of plant growth and senescence. Other traits that we may consider when as response to salinity is the ion balance. Everybody in the room know that ion balance, sodium, potassium are crucial in terms of phenotyping. But there is also some other effects, especially medium long term, concerning the nitrogen metabolism, which really salinity affects the nitrogen metabolism probably much more than water stress. And this may give us some ideas on what to phenotype, okay? But what is important is, as in other phenotypic activities, distinguish between physiological traits that are related with plant performance, growth, yield at the end, against the traits that are related with survival. Survival, for example, if you are analyzing the amount of reactive oxygen species as a trait for phenotyping, you are actually putting more emphasis on survival, which in most of the situations is not crucial. It's most important look for traits related with how happy the plant is growing, if the plant is delaying senescence, if the plant is keeping stomatans open, if the natural metabolism is good and the protein accumulation is adequate. So this is the point that I want to stress. And then phenotyping, what plant traits we have to phenotype? Just to summarize, plant growth, plant senescence, gas exchange, photosynthesis and transpiration, duality of processes, and metabolism, like nitrogen status, ionic balance, especially. Of course, they may be more, but at least my experience is concerning those two. In terms of phenotyping, phenotyping is a complex process that implies, that implies different actors, but I will put emphasis on platforms. Platform is from where the sensors are taking the information for phenotyping. And the second is, I will put also emphasis on sensors. Of course, we may discuss even on low-cost approaches for the data integration, spatial variability, etc., but there is no time. In terms of phenotyping tools, there are three different categories, basically two. One should be the remote sensing, proximal sensing and imaging, and the other should be the lab approaches. I will put emphasis now on proximal sensing and imaging. And of course, in terms of sensors, there is a myriad of low-cost sensors of different categories. Sensors related with temperature, with spectroradiometry, reflected by the plant, or with single digital RGB images. I will go further. In terms of platforms, of course, the range of platforms is huge, from ground platforms to satellites, but there are really low-cost platforms. We don't need to go to this kind of huge facilities costing millions of euros, or even to those phenocards, phenoshards that also very expensive, high throughput, but very expensive. We may keep our idea at the ground level, at the area level with low-cost approaches, and at the satellite level. I will show you some ideas. Ground level, if you have a phenopole, a selfie pole, that you may say pheno because you are using for phenotyping, with a camera remote controlled by Bluetooth, you are actually using a phenotypic platform. Here is an example in Zimbabwe doing some maize phenotyping with CIMIT, a collaboration with CIMIT. At ICBA in Dubai, here moringa tree being phenotyped, and here there is some quinoa plantlet also being phenotyped at the ground level, okay? With some aerial platforms. Of course, everybody has on mind drones, but nowadays they are low-cost, compact drones that have already embedded thermal, RGB, and multispectral cameras for few thousand of euros. Still, for somebody, maybe unaffordable, but I think it's quite interesting. And satellites. Satellites is a new revolution coming. There are a lot of satellites. Of course, we may consider the Sentinel-2 is Open access is free, using several spectral bands that allow the formulation of many different vegetation indices. The only limitation is that each pixel corresponds to 100 square meters, which is good for monitoring, but is not good for phenotyping. But there are also already commercial satellites, microsatellites like the Pleiades constellation that has a resolution not of 100 square meters each pixel, but 0.25 square meters, which is within the domain of phenotyping already, if you are using a small plot. But this is, of course, is not open access, it's not for free. Okay, so let's go to some, discuss a little bit on the tools for plant growth and plant senescence. We have, remember, growth, the green, and senescence, the red. We need to phenotype that. We have different approaches. I should put here also the satellites, because the sensors, but let's move to some ideas. Spectroradiometers. This is the classical. We may have different approaches from leaf sensors. Here is the dual X to the ground canopy sensors like the green seeker. Those measure basically vegetation indices related with biomass. With biomass on greenness. In any case, the light reflected by the plants gives us information on greenness, may also give information on photosynthetic efficiency, may give also information on the water status, depending on the wavelength. Okay? And of course, traditionally, for the last 30, 40 years, and even more for some of them, there has been formulated a large number of vegetation indices. Here there is just a few of them, but the number of formulations reached the dozens, I will say even hundreds at that time. Okay, one of the low-cost approaches for phenotyping at the ground level is Green Seeker. Green Seeker only costs 400 euros. It's quite good, especially for ground evaluation. But if you are going to lesser cost options, the vegetation indices formulated using RGB, red, green, blue, digital images are really an extremely good alternative. What you need is just to use a camera, a good camera, or even a mobile phone camera. And the idea is the RGB indices, even that they are more limited in terms of a spectral range, we have 3 different bands compared with the spectral response in the visible and the near infrared. But the RGB images has an excellent spatial resolution, and they are taking the part in the photosynthetic active radiation coverage, and they are quite straightforward for calibration. So if you compare RGB images with some traditional multispectral indices like the Normalized Difference Vegetation Index, NDVI, usually the vegetation indices formulated with RGB images performs in most cases better than the NDVI, and a lesser cost. Okay, and concerning the RGB images, the vegetation indices are basically information derived from the different ways the color is represented in a digital space. So there are different ways to represent the colors, RGB, the hue intensity saturation space, the CL lab, the CL loop. I will show you here. Sorry. No. It disappeared, sorry. Well, the point is that you may you may formulate those indices using open access software. We have our own software. Anybody interested, I may put in contact with a postdoctoral, and he will help you in not only in getting the software, but also in implementing. Software that, if you have RGB images, you may calculate different vegetation indices. Okay? I don't know what's happened, but it was here. So the idea is taking the images, and here you may see some of the color representation of different indices. Perhaps the most known are the green area or the greenest area, and also the normalized green-red difference index, etc. Those indices are really good indicators in many cases. Here there is an example of the long-term date palm trial at ICBA. We did a collaboration with them, and this trial has been for more than 20 years now, and taking images just as of the canopy for each individual date palm tree, we got a quite reasonable correlation in terms of fruit yield, number of branches, total trunk height, or trunk diameter, so, which are indicators of biomass. So using different vegetation indices, okay? But of course, machine learning is paving or is helping to take fully advantage of this kind of information. Here there is one example of Gabriel and Mark. They did the work, well, I took advantage in Frontiers in Artificial Intelligence. They evaluate through an aerial platform individual tomato plants. And what they found is a good prediction, especially when the fly was closer to the harvest, which is actually a good demonstration on potentially low-cost approach that really is working well. So the prediction was good. The error, especially on the last flights, were really low. But even we may go one step further. We may forget about vegetation indices. Take only the RGB images and use in a fully empirical way through machine learning. And this is, for example, the case of many mobile phone apps devoted to evaluate plant disorders, to recognize automatically what plant species is. Here you have several. I will recommend Plantix. It's quite consolidated from, and also we are developing with ICBA and Dr. X NAVAD on Google Play that for recognizing disorders and different horticultural plants. So this is really getting the images, forget about formulating indices, and try to relate the images with the recognition of one disease or whatever, if of your interest. RGB images give for much more. Not only counting, for example, spikes or plantlets or trees, but also to monitoring, or also to do three-dimensional reconstructions. Here, for example, there is one, you may recognize diseases, you may count in the number of kernels in maize, you may count in the number of spikes in situ, you may get an idea of the plant height. Here there is one example in sorghum. So a comparison of three-dimensional reconstruction, 2 minutes. That is giving quite good prediction. Gas exchange, some ideas on gas exchange. Gas exchange, when I'm talking on gas exchange, many people will think about air gas and porometers, but there are other options for high throughput phenotyping, like infrared thermometers, few hundreds of euros for a good one. Here there is one example on date palm that also is predicting a yield. But in the market you may find another approaches for thermal cameras. Here there is a mobile. For the last 5 years there are mobiles combining RGB and thermal cameras with that may use in different modes with the thermal and embedded the temperature, or with the RGB and embedded the thermal. And here there is some example, a thick again, moringa, trees being assessed with this kind of mobile, or there are other options like thermal and RGB images with the duo FLIR. The cost of those cameras are really quite affordable. The mobile phone is not more than 500 euros, and this should be a little bit more, 1,000, 1,200, but give a very good information. Other traits, I am finishing laboratory traits. I'm putting special emphasis on a stable isotope signature. Why a stable isotope signature? And because especially carbon isotope composition is a long-term integrated indicator of water status, especially of stomata aperture. Here you may apply in crops, most of crops, C3 plant species especially, and all the trees species. And the idea is the more close is the stomata, because the salinity stress, the carbon isotope composition, delta minor, will be higher, so less negative, and the opposite. The better the water status, the more negative will be the carbon isotope composition, or the higher will be the discrimination. That depends on how you express. Some examples with in Tunisia to relate durum wheat genotype with carbon isotope discrimination composition, again grain yield. Also in date palm, indicating the better the water status, the more negative, and the higher the more negative the carbon 13 and the more negative. Salicornia is another example. Here it should be, you should take carbon isotope composition in the opposite side, because it's a purely halophyte. And just to finish some other traits, one example of ionic balance. Here is IGBA again. There is the agronomical procedure is to put 40 tons of manure in the soil, because it's sandy soil. And that manure is very rich in magnesium. What's the consequence? Even on the so-called freshwater treatment, the genotype the most affected in terms of biomass and grain yield are those exhibiting higher contents of magnesium. So the manure is really producing salinity, even in the best of the scenarios. That's a good. And nitrogen 15 and total nitrogen content is an other option. Here there are some other studies in Tunisia showing some nitrogen 15 and total nitrogen content, and here you may have in date palm, there are some examples in durum wheat under control conditions. Yeah, anybody may say it's expensive. Carbon isotope may cost 8 euros, carbon total nitrogen content, carbon isotope composition, and nitrogen isotope composition. Otherwise, you may do NIRS, you may calibrate whatever you want to see, and the cost will be 1, 2, 3 euros per analysis. So the conclusion is physiological yield components are the adequate, are the right traits to phenotype. Low-cost phenotypical approaches that exist. I will especially put emphasis on RGB images, and remote sensing tools are not the only alternative. Okay, and thank you, and that's all. And well, sorry if I took. No, yeah, I know that it's still on time because we started. Yeah. So.
Thank you.
Thank you. I don't see there is time for a question, or we should move to.
Well, I believe we'll give the floor for one question because of time, and okay.
Thanks for your presentation. Nice presentation. My question is about.
Yeah.
We can say, okay, stomatal conductance is a response.
Yeah.
It's a response of stress.
Yeah.
But is there any way to measure the stomatal conductance to see which stress? Because we can have salinity, we can have high temperature. You have this boundary layer. It's too complicated to see.
Yeah, I mean, good point. Normally, what you realize is that the plant which close stomata are affected by a given stress. Drought stress, salinity. Of course, the important point on my view, especially on phenotyping, is that you are, with those techniques, you are comparing different genotypes together. And then, regardless the temperature is higher or lower, all the genotypes have been exposed to the same conditions. So the point is not to define what is the happiest genotype in terms of keeping stomatals open. Regardless that the final reason is due to salinity or not.
To distinguish between
Yeah,
if the stress is salinity
Well,
or is high temperature.
To distinguish, yeah, high temperature sometimes is a combination of both. But at least what you have is, but if you are using a gas exchange, a light core or a porometer, what you will get is the same. I mean, a value of the stomatal aperture on the photosynthesis, but you will not have clear conclusion is heat, is salinity, or is the interaction between the both.
Okay.
Okay, and with the advantage that the porometry or gas exchange is not a high throughput as for example, this kind of techniques.
Thank you. I guess with constraint of time, it's very interesting topics. I thank Dr. Jose for his presentation and this topic on relating the phenotypic threat to the water or salinity resistance and resilience. If you have more questions and clarification, Dr. Jose is with us here this morning, and you could ask him or questions. So I believe we go straight to the second presentation. I hope that colleague Murat Aykam is with us online. The following presentation is on the determination of salt-tolerant wheat cultivar by Physio, Biochemical and molecular approaches at Seedling stage by Murat Aykam and his collaborator Marwan Baslam, Toshiaki Mutsi and Mustafa Yildiz. Murat is going to present.
Yes.
Okay, the floor is yours.
Could you see this slide? This is Murat from Nigapur University. Our title is Determination of Salt-Tolerant Wheat Cultivars by Physiochemical and Molecular Approach at the Seedling stage. As you know, the salinity stress is going to more damage to plant yield in agricultural fields in many species. In this study, basically, we make a screening which the cultivars commonly used by farmer in the field in the Middle East, and Russia and Ukraine cultivars under stress, under such stress conditions. In the study, we aim to evaluate and screen at the seedling phase at the salt tolerance of 16 commonly cultivated cultivars. And second point is determine and cluster the salinity tolerance related traits markers by deep learning approach for improving future wheat breeding studies, and evaluate the salt tolerance mechanism of salt tolerant wheat cultivars. As you can see in the slide, we use 16 cultivars, and these cultivars belong to different countries, but these all wheat cultivars is winter type and bred wheat cultivars. And the seedling in the plant's first 10 days growth under normal condition, from 10 days to 20 days in two groups. One group in the control condition is continuing, and the second group is the salinity stress condition, and for 10 days. And after 10 days, we make the measurements of germination. The first stage is the germination stage, second stage is the seedling stage salt stress. In the germination stage, in the germination stage, we take germination percentage, germination speed index, seedling vigor index, and the seedling stage, fresh weight, dry matter, seedling height, root length, salinity tolerance, chlorophyll content, proline, melandoaldehyde, and antioxidant enzyme. And later we check the tolerant and sensitive culture based on three stress marker gene in wheat. You can see the result. We screen the all cultivar first germination stage, and we evaluate. In the germination stage, we find cultivar 4 and 6 more tolerant, cultivar 10 and 15 is sensitive in germination speed index, seedling vigor index, and especially salt tolerant index parameter.
We don't see your presentation.
You can see?
It was on the screen, but we lost it.
I will try again.
No, we can hear you, but we don't see the presentation.
Okay. Now you can see?
Yes. Great.
Okay. In the germination stage, we find two tolerant cultivar and two sensitive cultivar, which is cultivar 4 and cultivar 6, cultivar 10 and cultivar 14. 4 and 6 as tolerant, 10 and 15 is sensitive in germination speed index, seedling vigor index, and salt tolerant index parameter. At the seedling stage, we check classic salinity tolerance trait, which is commonly used in the plant evaluation for salinity stress. And the seedling height also, we find 4 and 6 tolerant, 10 and 15 sensitive. Root length, same cultivar show tolerance and sensitiveness. Fresh weight and dry matter also show 4 and 6 cultivar, tolerant, 10 and 14 is sensitive. And later we check the chlorophyll content, which is related to photosynthesis and biomass, and basically. And these parameters also, chlorophyll A, B, and total chlorophyll content also, cultivar 4 and 6 show high chlorophyll content, and cultivar 10 and 15 shows lower chlorophyll content. And later we check melandoaldehyde and proline content. As you can see in the figure, again 4 and 6 show less MDA content, which shows the cellular damage is less under salt stress condition, and 10 and 15 show higher melandoaldehyde content, which shows the plant cells get higher damage from salinity stress. And proline also is the osmolyte, which is the show salinity tolerance, one of the salinity tolerance parameters in the studies. Cultivar 4 and 6 shows higher proline content, and 10 and 15 shows lower proline content. And later we check the enzyme activity, which is CAT, salt, and glutathione reductase enzymes. Also enzyme activity, also these two, 4 and 6 cultivars show higher enzyme activity under salt stress. 10 and 15 number cultivar shows lower enzyme activity under salt stress condition. And later, to evaluate all of the data together, we make multivariate analysis and hierarchical clustering. As you can see in the slide, sensitive cultivar make one group, and tolerant cultivar make one group under salt stress condition. This line below is the salt stress condition, upper is the control condition. Control condition is not specific grouping, but under salt stress condition, we can see the tolerant, sensitive, and moderate cultivar as 3 groups. And later, in the heat map clustering analysis, also we could see the control and salt stress exposed plants differentiation, but sensitive cultivar under salt stress condition group on the top, and salt tolerant cultivar under salt stress condition group on the bottom line, which shows the huge differentiation among the trait results. And later, to compare these traits, which is correlated each other in 2 tolerant cultivar and 2 sensitive cultivar, and we find same clustering in here also. Glutathione reductase, superoxide dismutase, catalase, and proline show higher correlation in both cultivar, salt tolerant and salt sensitive cultivar, but MDA show less correlation between the other traits in salt tolerant and salt sensitive cultivar. And later, to check these 2 salt tolerant and salt sensitive cultivar with the 3 molecular marker genes, which is HKT1 is related to sodium transportation in the wheat, P5CS, which is related to proline synthase, and DHN, which is related to dehydration in the plants. As you can see, intolerant cultivar has higher activity under salt stress condition, but sensitive cultivar has less relative transcript level of marker genes under salt stress condition. As the conclusion, we classified 16 commonly cultivated wheat under salt stress condition. Among the 16 cultivar, we find cultivar 4, which is the commercial name is XJ96, and cultivar 6, which is commercial name is Demir 2000, is high salt tolerance, high salt tolerant cultivar. Cultivar 10, Tolmen 01, and cultivar 15, which is Esperia, which is salt sensitive cultivar. And tolerant cultivar display lower germination rate and growth parameter reduction and better biochemical and molecular changes, result in IB identification of the proline and Ross-Swanger pathways in the process of tolerant response to salinity in the early growth stage. Collectively, the cultivar-specific stress response traits identified in the present study act as the valuable resource for the research interested in plant responses to salinity at the seedling stage. Additional studies will be necessary to unravel the mechanism of resistance to salinity at reproductive stage and to use identified tolerant culture as donor parents to develop potentially high productivity wheat plants for the crop breeding under salinity stress condition. Thank you very much.
Thank you. Thank you for your presentation. And we have some time for a question or two. If anybody from the audience or from colleagues online. Okay, we have one question from...
Yes, please.
Thank you very much for the presentation. I will just ask you if... Go ahead.
Thank you for the presentation. I want just to ask you, is the seedling stage of wheat the most sensitive stage, or can we have some changes later on, and especially when wheat start taking more water? Thank you.
As you can see, in the wheat, the seedling stage is the most sensitive stage for salinity stress. And also based on other research experience, if the seedling stage plant can survive under such stress condition, and reproductive stage is more good. We can see, actually the reproductive stage plant is more self-tolerant than seedling stage. If plant can survive in seedling stage, we can have more option to get high yield in the reproductive stage.
Okay, thank you. I guess, thank you, Dr. Murat. Now we'll give the floor to Professor Yasser Al-Salama, who's going to talk to us about the effect of seed priming with zinc oxide nanoparticles and saline irrigation water in yield and nutrient uptake by wheat plant. So Dr. Professor Salama Yasser, you have the floor. Can you share your presentation? Okay, you put it on presentation mode. Okay. So you have 15 minutes, Professor. You are muted. Still, we don't hear you. We can see you, but we see the presentation. We see you, but we don't hear you. Do you have any problem, Professor? We don't hear you.
Now is okay?
Okay, now good, yeah.
Yeah, okay.
Okay, go ahead. Could you put your presentation on a presentation mode?
Okay. Now is okay?
Not yet, but maybe it's coming. Yeah, that's good.
Okay. Good morning. Firstly, I would like to thank the scientific community and organizing committee to take me the opportunity to participate in this important forum about salinity. And I am speaking from the Syria, representative to the Oxide, the Arabic Center of for the studies of arid zone and dry land. And I am a professor in practical agriculture at Furat University in Syria. It is well known fact that world resource of the fresh water are getting exhausted through the increasing demand to meet the ever increasing requirement of the increasing the world population. Therefore, the use of the low water quality as the groundwater, water, and treated seawater should be considered as a complementary source for the agriculture development. Wheat is the moderately tolerant as a background for the wheat plant to salinity stress. It is considered either to the drought and the major biotic stress that affected the production capacity in the arid and semi-arid. Seed germination and seed establishment are the most sensitive stage to the salinity, and the salt stress can adversely affect seed germination and initial seedling, both by both the osmotic effect and ionic toxicity, which will include the oxidative stress. What about the nanotechnology in the agriculture? We did a similarly about using nanotechnology in the agriculture. In the main, we need it in the crop improvement as the nanobiotech technology, as the nanotiller or transgenic planet. And the other side, we make it a group production as the nanobiotics, or the group growth as nanofertilizer, or the soil enhancement as the nanomaterials, and in the precision farming as the nanosensors. Finally, we use the nanotechnology as in the stresses tolerances as the nanoparticles in the dirt or the salinity or the UV stress. Now we will use it, the nanoparticles, as to meet the stress tolerance as salinity. The use of nanotechnology for seed brimming is a new area of research. Seed brimming with the nanomaterials can promote seed germination, enhance initial vegetative growth, and it is a simple and cost-effective, and brimmed seed can rapidly absorb it and renovate the seed metabolism, resulting in the higher germination rate and degrees interest physiological and uniform in the germination. Seed brimming can also improve plant growth and production capacity of groups. Different methods are used to apply the zinc oxide nanoparticles on wheat, like flower spray, soil mixing, and seed brimming method. Zinc oxide nanoparticle at low concentration promotes the production and antioxidant, enzymic and non-enzymic moleculars and phytohormones and another in the overexpression of the new water canals. Further improving the absorption of water and nutrient with zinc nanoparticle seed brimming can help the initial growth of seedling. And new letters show that brimming of the wheat seedling with the zinc oxide nanoparticles, 50 milligram per liter, and after 20 days recovery of the plant were exposed to the salt effect, about 20 millimole in sodium chloride for 10 days, it was found that greening reduced the concentration of the sodium and increasing the weight uptake, the water uptake. However, when plant exposed to the salt stress, the nano-zinc nanoparticle print plant had significantly higher photosynthesis, higher stomatal conductance and transpiration rate comparing with the non-primate plant. The main goal or the main objective from our work was to study the influence or the influence of the effect of the seed greening with zinc nanoparticle and saline water irrigation, downed water on yield and nutrient uptake, nitrogen, phosphorus, potassium, and zinc of water weight variety Oxide 1100 grown in calcareous soil. Two groups of the material of the and method, two groups of weight variety seed, triticum, as I see the Oxide variety, were used. The first one was left without any treatment and as the control non-primate seed. The second group was primate with 50 milligram per liter ppm zinc nanoparticle for 4 hours before sowing. The zinc nanoparticles is characterized as the particle size less than 50 nanometer were obtained from the Big Brock directors, University of Oxford in United Kingdom. Both groups were used in the pot experiment and the 4 level of salinity. The pots were filled with 10 kilogram calcareous soil, sandy loam, calcium carbonate about 24.5, and the salinity about the EC about 2.27 decimins per meter. The primate and non-primate were snow separately in each pot. The seedling were irrigated with tap water as a control, about 0.52 decimins per meter as a control, and 3 level of salinity ground water. W2 4.42, W3 6.82, and W4 9.13. This is after 160 days from the planting. Plants were harvested and the green yield per pot and straw per pot, and the 1,000 kernel weight were recorded. Representative planted sample were digested for nitrogen and phosphorus and potassium and zinc determination. And finally, the SPSS software was used for statistical analysis. The main result and the important result in this study was that we can see that the increasing of the salinity of irrigation water significantly decreased all the studies trial in both primate and non-primate seeds, but reduction was significantly lower in the primate seeds. Increasing water salinity up to 4.42 and the second level and third level caused remarkable decline in the green yield of the non-primate weight seeds by the percentage about 13 and 20 and 40 representatively to the level of the salinity. And seed brimming with the nanoparticles increased green yield about 36 and 24 and 30 representative to the level of the salinity water. Concerning the steroid and the 1,000 kernel weight, that obtain the similar trend of the green yield. About the effect of the bringing in the nanoparticle in the nutrient uptake, macronutrients, nitrogen and phosphorus and potassium uptake by green and sorrow of wheat plant were significantly affected by salinity in the post-treatment primate and non-primate seeds. The maximum uptake value by green value were obtained in the W3 level of the salinity water. It has been noted that the seed priming significantly enhanced the uptake nutrient by green component with the non-primate one. Similar trend was recorded for nutrient uptake by the stolo at the same time of the nutrient uptake was the least at the highest salinity level for the post-treatment. This could be attributed to the effect of the higher soluble salt on decreasing the water potential between the soil solution and the root cells, developed by the decreasing the water flow and absorption, which will absorb the effect in the nutrient uptake. In addition, the effect of the higher concentration of the sodium and chloride on the uptake of the other beneficial cations and anions restricting in the nutrient uptake to extent that the deficiency take place. This may be due exclusive salinity has detrimental effect on the growth and weight yield by the possibility that plants grow under the saline. Okay? Okay.
He left.
Okay.
Okay, we still, yeah, we can.
Conclusion.
We just lost
Conclusion.
you. We just lost you for a while. Go ahead.
Yeah, okay. Salinity stress as well to effect have on the weight, plant growth, and green yield and yield components. Seed priming with zinc oxide nanoparticles was an effective technique to improve the performance of the weight plants, thorough and salinity, and the stress conditions. Thank you for your attention. I'm finished. I'm ready for any question.
Thank you, Doctor.
Thank you for you.
We have a floor for one questions. So Dr. Salama has talked to us about the importance of priming using zinc oxide to improve performance of durum wheat on salinity and growth.
Okay.
So from the floor, we have one hand raised.
Yes.
Yeah, go ahead.
Thank you very much. I'm Johnny from Nigeria. I'm Dr. Asma Vekana from Benue State University in Nigeria.
Okay.
I actually enjoyed your discussion about the priming using a zinc oxide. I really want to find out, are there any special benefits of using zinc oxide as an important ingredient for seed priming, or there are other nanoparticles as well that we can also make use of them in the priming of seeds? Thank you very much.
Okay. We can share another slide here. You see that? See this slide?
Yes.
Yes. In this slide, we can understand how the nanoparticles, as zinc nanoparticles or anything that the nanoparticle affect in the seeds. The main effect is in activation the stress of genes, and then in the make damage in control and make repair the damage that retained from the more salinity. And maybe the same thing happened in ionic and osmotic human by the SOS pathway. All of this for the sorrow or the drought or the salinity may take place from when I bring it the seeds in nanoparticles and make the activation for the roots and make root take place and to be more able to find in the salinity conditions. I hope you're okay.
Yes, I understand, but just one more question. I don't want to waste your time. Do we also have other possible metal oxides, nanomaterials that can as well be used beside zinc oxide nanomaterials?
Okay. We have another treatment in the project, but we may select one for this floor.
Okay.
Okay, thank you.
Thank you very much.
We can connect with us
constraint.
and explain that. We can connect with us and explain that.
Okay, with time
Thank you very much.
constraint, I guess we have to move to the next presentation. Thank you, Professor. And
Welcome.
so
Thank you for
thanks. We'll move to the next presentation. Will be presented by Dr. Shuriti Pingle on the effect of salt stress on proline content in corn crops. Are you with us, Doctor?
Yes, I am.
Yeah, I would just ask you if you could take a few minutes or 1 minute to present yourself to the audience.
Yes, I will, but since this project was performed by 2 persons, so will it be okay to present here with my partner?
Yes.
Thank you.
Yeah, you could start. I'm asking Dr. Yasir Salama to unshare his presentation. Okay, thank you.
Thank you. Okay.
So, Dr. Shuriti?
Yes.
Okay. Go ahead.
And can you everyone see my presentation slide?
Yeah, just if you could put it on a presentation mode. Put it on a full screen.
Yes, sir.
On full screen. Not yet. Hello? Yeah. Hello?
There seems to be a problem. I cannot put it on a full screen mode.
Yeah, at the bottom of your screen, there is a kind of a screen, small icon. You just...
At the bottom of my screen?
Yeah, at the bottom where you have this, when you add the, you improve I mean, there are some icons at the bottom left or the bottom right of your screen.
Yes, sir.
You have like one button there too.
Oh, we cannot find that.
Okay, you could go this way.
We could, okay, thank you.
Yeah. Okay, the floor is yours.
Can you see next slide?
Yeah, we see your slide. You just move from one screen to other yourself.
We will try again.
So you have 15 minutes. Don't lose too much time on this.
Yes, sir. So our topic is on salinity in maize. We have to see the effect, salinity effect on proline in maize.
Yeah, go ahead, go ahead. Go to the second slide, third slide.
Traditionally, its growing area was more focused on Bihar, Madhya Pradesh, Rajasthan, and Uttar Pradesh. To some extent in maize area, including Karnataka and Andhra Pradesh, it is also very popular in the low and mid-hill areas of the western and northern regions. Soil salinity is a key challenge in several regions of India, such as Gujarat, Uttar Pradesh, Maharashtra, West Bengal, Rajasthan, and Odisha. Salinity stress affected plant growth and productivity, water relation, photosynthesis, and carbon assimilation, grain development, and filling. Proline is the most common indigenous osmolalite accumulated under various abiotic stresses, including salinity. Next page. The role of proline in cell osmotic adjustment, membrane stabilization, and detoxification of injurious in plant exposed to salt stress is widely reported. Thus, the objective of the study was to study the effect of salinity on proline content in maize. The experimental design was CRD, complete randomized design. We applied 6 levels of salinity, 0, 50, 100, 150, 200, 250, V millimolar, and for each with 5 ml solution for 3 days, a total of 12 days. For a method, we have used beds method. We took 0.2 grams of fresh plant sample, homogenized with 3% salicylic acid, centrifuge at 300 RPM for 20 minutes. Then we have extracted the precipitation, then given the further treatment according to the beds method. We have collected data on germination rate, germination percentage, number of leaves, shoot length, leaf color, days of maturity, weight of seeds at the day of sowing. For result, we have, through the experiment, we found that 2 varieties that we have used, Advanta Peka 751 and Sri Janta 7710, which are available in India, are very yielded very good result under stress condition. These are some of the results graph that we have presented here on proline. Germination percentage, we can see the 2 varieties where they have yielded maximum yield. Same for the rate of germination. For conclusion, in this study, the effect of applied NSA on growth and proline concentration of maize varieties were investigated. We have concluded that there was significant increase in growth and proline concentration of maize varieties. With our study, we can help farmers who are suffering from soil salinity. Here, we use the method for proline accumulation described by Bates method on different types of crops. Thank you for your time, and we are extremely sorry if we have made any mistakes.
Thank you. Thank you. It was on time. in spite of the waste of time at the beginning, which is good for us to just save some time for our session. Thank you again, and I will just...
Sorry for that.
No problem. We just, if any question from the floor or from colleagues online. Apparently, I don't see any hand raised. Okay, we have Professor Jose.
Yeah, many thanks for your presentation. Just,
Wait, I'll give you a question.
microphone, I think. Just
that guy who has to put it on or off. Where you keep it.
okay. Just a first question. Do you think proline is then a positive? High accumulation of proline is a positive trait or the opposite? And why?
Did you hear the question?
No.
Okay, the question was, does the proline rate is a positive or a negative indicator? That's correct.
Proline rate is positive indicator for salt stress.
Yeah.
In fact, it is positive indicator for any biotic stress.
Okay, leave it.
Thank you.
Questions and answers to questions. So we're going to move to the next presentation by Asma Omsat. I hope I pronounced it correctly.
Yes, it's Asma Omar.
Okay, which is on the effect of different irrigation water salinity levels on nutrient uptake, biochemical content, and growth response of blue panicum, quinoa, and silage maize. So the floor is yours. I will ask Shuriti to unshare the presentation. Oh, okay, so that's all. This is your presentation, right?
Yeah, yes.
Okay, go ahead. Put it on the presentation mode if possible. Yeah. Yeah, that's good. Okay, go ahead.
Great. Hi everyone. Today I'm going to present a presentation about the effect of different irrigation and water salinity level on nutrient uptake, biochemical content, and growth response of blue panicum, quinoa, and silage maize. So this study is a part of a large agroecological mega project of ENA Moroccan Medium Terms Program, which aims the development of sustainable solutions to manage and buffer the effect of salinity on the soil of irrigated area in marginal lands, of course, to improve their productivity and upscale the output. So, like we know, the study was conducted in Sus-Massa region, which knows for the semi-arid climate. This climate is characterized by low rainfall to maintain crops and by a strong evapotranspiration that accelerates the process of degradation and salinization of the soil. This leads to an increase in salinity of the soil and irrigation water. Of course, the soil salinization is a limiting factor of agriculture productivity. Scientific study selected a range. Scientific study selected a range of salinity tolerance crops, which knows new salt-tolerant alternative crops that adapt well in this semi-arid climatic condition. Alfalfa can be grown and produce a harvestable yield in saline soil and watered with salt to water. In this study was tested blue panicum, quinoa, and silage maize as salinity tolerant crops. As we see, they are all classified in group of allophyte species, but they different in adaptation under salinity condition. For example, silage maize is the soil salinity tolerance threshold value is 1.8 decimals per meter. For blue panicum, is high salt tolerance species, which carries irrigation with water salinity up to 15 grams per liter, and it is also resistant to drought stress. Also, quinoa, just the threshold exceeding is 9 decimals per meter, and it has the ability to resist drought stress. So the purpose of this study is to confirm the result of previous study, efforts by comparing the tolerance of blue panicum, quinoa, and silage maize to different salinity doses, and to understand the mechanisms of the effect of salinity on the absorption of minerals. Understanding this effect will allow the development of fertilized strategies that are suitable to improve the yield. So to do this, we have conducted the experiment in the experimental greenhouse of the Regional Center of Agronomic Research of Agadir, INRA, and we have germinated the seed. In the trays filled with peat and irrigated them for 20 days by just watering with tap water. And after germination, the seedling were transplanted into mesosome filled with peat and irrigated regularly with different saline water treatment. So the experiment design was a randomized complete block of 4 treatment and 4 replicate for each treatment. The applied treatment were control or a treatment 0, that is tap water, and 3 treatment of electrical conductivity of 3, 6, and 10 decimals per meter. After 8 weeks of growth, the plant of all species were harvested, then separated for their leaves, stem, and root. So the physiological, biochemical, and mineral parameter studies during this experiment were stem height, above ground and root dry biomass, potassium, calcium, and sodium content, proline content, and chlorophyll content. And we have conducted statistical analysis using SPSS software. Data are analyzed using ANOVA and 2K post hoc test. So we go to results. The 3 graphs show the mean of stem height, above ground, and root dry biomass of the 3 species by tested treatment. We found that the stem height and above ground dry biomass of blue panicum were not affected by the applied salinity treatment, except for the root dry biomass, which decreased at 10 decimals per meter compared to control. And that is the same what has been observed in many studies, like Coiro et al. In 2013 and Atia in 2019. For quinoa, increasing irrigation water salinity only decreased plant growth and root dry matter biomass at 10 decimals, without a significant effect on the above ground dry biomass, compared, of course, to control. Similar results were obtained by Harish, who reported that quinoa is considered a facultative halophyte with a salt tolerance threshold equal to 9 decimals per meter. For silage maize, we found that the physiological propriety was decreased with the increase in irrigation water salinity compared to control. We pass to the other physiological biochemical property that were the photosynthetic pigment content of the three species by this tested treatment. We found that carotenoid content in blue panicule increased significantly with increase in irrigation water salinity compared with the control, while showing no effect in chlorophyll A and B. Also, we found that quinoa showed no significant effect on the applied salinity treatment on photosynthetic pigment content. Instead, silage maize, the increase in irrigation water salinity resulted in a significant decrease in chlorophyll A, B, and carotenoid, which could be caused by the high enzymatic activity of chlorophyllase, which increase the sodium concentration. The following graph shows the mean of proline content of the three species by tested treatment. We found that there were no significant effect of the applied salt concentration on proline content for all crop species. The graph shows the need of calcium content in leaves, shoot, and root of the three species for each treatment. We found that calcium uptake at leaf and root in blue panicum increased with increase in salinity levels compared, of course, to the control. For quinoa, there were no significant difference observed in calcium content. And for silage maize, the calcium uptake decreased with increase in salinity level to be significant for irrigation water with an electrical conductivity more than 6 decimals per meter. The other graph shows the mean of potassium content in the leaves, shoot, and root of the three species for each treatment. There were no significant difference observed for potassium uptake in blue panicum and quinoa between treatments. This finding consistently with the result reported by Roman, which shows that quinoa plants are able to maintain the uptake of potassium despite high sodium concentration in the soil after extended salt stress. For silage maize, we found that potassium decreased at leaf and the root part compared to control. The last graph is show the mean of sodium content in the leaves, shoot, and root of the three species. We found that sodium uptake increased with increase in salinity in both crops. Also, silage maize showed a significant higher sodium content than blue panicum and quinoa. This would be attributed to the ability difference of both crops to involve both mechanism, exposure, and the comparative minimization of adaptation to salt stress. We conclude from our results that increasing water irrigation salinity did not affect the plant length, above ground dry biomass, and proline content for blue panic, and also did not affect on above ground dry biomass, photosynthetic pigment, proline, calcium. While in silage maize, salinity doses affected all parameters measured except proline content. We can see that the mechanism of salt tolerance varies between species, like for example, blue panicum based on high uptake of calcium and potassium to resist high salinity. Quinoa based on maintaining calcium and potassium uptake despite high sodium concentration, and silage maize based on high uptake of calcium up to salinity threshold that is more than that is more than 6 decimals per meter to resist high salinity. So using Lupaniculum and quinoa as alternative plant could be a beneficial approach to valorize salt-affected area, overcome the salinity problem, and improve the income of local farmers. Thank you for attention. If there is any question, welcome.
Thank you. Thank you for your presentation and mainly your final conclusion that blue panicum is good crops for saline region. So I just open the floor for a question. Yeah, we have one question from.
Yes.
Okay, just. You have one question. You will hear it right now. أجي هنا أجي.
Yeah. Thank you for your presentation. My question is about maize, silage maize. Yeah, we can notice that for calcium and potassium are higher in silage maize. However, that is the more sensitive species. How do you? Normally, it must be the contrast, especially for potassium and calcium that interfere with sodium.
Yes, well, I found when I do research, I found that there is a contrast, but it can be caused by that I didn't let him
there were more potassium in silage maize than blue panicum and quinoa. And also the same case for calcium, that are both criteria for salt tolerance. But in your case, silage maize is the more sensitive species.
Maybe it's because it's just a result for 8 weeks of drought. It didn't come to a level who will absorb more potassium. And also every species have a particular mechanism.
You could check only for potassium-sodium ratio. That give you the, You could check for sodium-potassium ratio. That is a criteria for tolerance.
Yes.
Thank you.
Okay, thank you. I guess we have. Thank you, Asma, for your presentation and your presence with us.
You're welcome.
I will move to the next presentation on allometric models for predicting root biomass on field crops in the salt-affected clay soils, which is a novel approach. Presentation will be given to us by Amrah Aymamidov. So I hope I pronounce your name correctly. If you are online, just the floor is yours.
Yes, I am online, and thank you. I am going to upload.
Okay, if you could just present yourself quickly for the audience and then go ahead for the presentation.
Yeah, I am Aymamidov, and I am my affiliation is with Doctor University Japan and Institute of Botany Azerbaijan. And I am mostly soil scientist, but in team we work with crop physiology too.
Thank you. Thank you.
And may I upload? I am sorry that I lost my voice because of common cold laryngitis, but it should be okay. I am sorry. And I am also after exam. That's why I was a little bit late. I am sorry. Yeah. So can I upload or there is ready man?
Yeah, you can start, of course. Share your presentation and the floor is yours.
Yeah, yeah. I would like to ask if everything is okay.
Yeah, we see it. Put it on the presentation mode and go ahead.
Presentation mode. It looks like we have some kind of problem with presentation mode.
In the bottom left side of
Yeah,
the
yeah, it's just
screen.
button there is standard presentation mode, but why?
Yeah,
it
down,
cannot-
down. Take yours. No, that one, yeah.
Yeah, it's true, but sometimes because of high uploading, it does not work properly. I start with that. Okay, thank you. So may I start?
Yeah,
Professor?
go ahead.
Okay, again, sorry that after exam and after common cold Fahrenheit, I lost my voice a little bit, but you can stop me anytime. I will be happy to re-answer or re-present. Okay, allometric model to predict root biomass of field crops. So the work was done in different countries, but it's associated mostly with Totori University and Institute of Botany, Academy of Science of Azerbaijan. Totori University belongs to Japan. Okay, so root biomass, why we learn it, background? Because it is very important for crop performance and soil properties, by other way to alter, modify soil properties. Root biomass, root development is very important. It's important for carbon sequestration, soil quality, water and nutrient availability, microbial and enzyme activity, microbial community structure, plus soil physical quality like aggregate stability, hydraulic properties, and resistance against erosion. So it's important in arid and semi-arid zone, particularly in order better to get nutrient and water from deep layer. So plant development and distribution of photosynthate to root, stems, and leaf are highly affected by environmental factors and ontogeneous traits. In arid and semi-arid regions, environmental abiotic stresses.
Are you moving your slides? We are just still on the first, the title one.
Yeah, I am moving now. So to study root biomass is important for climate change and adaptation and mitigation. So, but to study root biomass is difficult issue. So a limited relation between plant organs should allow us to find relationship between plant organ and hence have to predict root biomass. It was our aim. So because for trees there is such allometric relationship that we saw that such allometric relationship also available for field crops. So it means that it could help us to mitigate, to make easy to calculate, to predict root biomass for our future work also. Yes. So objective was to develop allometric model to predict root biomass of crop species using root collar diameter. One of the easy measure in field condition. So we decided
Dr. Mamedov,
yes.
your
Yes.
screen is not moving for us.
Sorry?
You are still on the title. We see only the first slide. I don't know where you are in your presentation.
I am on the search slide.
Yeah, we don't see that. You have to move.
Yeah, I am moving. You don't see it?
No, we don't. We are. I mean, our screen is stuck on the first one.
Now, do you see second?
We still see the first. I mean, the first slide. Unless if you could unshare and share again.
Okay. Now I return back to first one.
You are in the
Do you
first.
see second?
Yeah. Okay. Now it's moving. Okay. Go ahead. But if you could put
Okay.
it on the presentation mode, will be better.
I agree. It looks like this presentation mode, we have some stuff.
Okay. It seems it is on presentation mode on your computer, but not on our screen.
Yeah. I changed to normal mode, not presentation mode, because of it's stuck. I understand.
Okay, that's good. That's good. Let's go with this one. Just move your slides.
Yeah, I am sorry that it's happening, but I am using, okay. So the aim of our work to predict root biomass using collar diameter. Easy field measure. Why we decided because of for trees, for woody species, such relationship are available. We thought, why not to use it for field crop, make our life easier from that point. So root biomass are very important from point of nutrient water availability and soil quality. In general, the root and root biophysics are associated with sustainable agriculture. So our objective was to develop a allometric model to predict root biomass of crop species using root collar diameter, one of the easily measured crop parameter. Next is methodology how we did it. We use 3 years experiment in semi-arid field. We use a plot, small plot, a relatively small plot, 40 to 40 meters, seedling standard. We use it was a clay soil, swelling, salinity level was very heterogeneous, irrigation level was traditional 70% from standard irrigation. Also, we use monolith excavation to excavate root and then to use washing method to clear it. And what crop we use? Alfalfa, sorghum, maize. And they have different root architecture to figure out if we can use our hypothesis that all 3 standing field crops, root biomass can be calculated from their collar diameter. So this is results. Collar diameter is related to the diameter which is in the border of roots and stem. And this is result showing that so different plants had a different root biomass, collar diameter, and collar diameter usually is associated with root biomass in our cases. It was true for all 3 plants with tough, fibrous, or adventurous root types. Next, this is alfalfa. It is small and root mass is small. Sorghum, average. Sorghum is much higher than alfalfa, but less than close or less than maize. Same order we had for collar diameter. So this is a relationship between root dry biomass and collar diameter. We find that between them there is linear or power function between collar diameter and root biomass. But power was better describing better root biomass depending on collar diameter. And here we have results. You see them for all 3 field plant species. And also you can see that root to shoot ratio and root biomass, they have some kind of same order, collar diameter. What does it mean? It means that selection of crop functional traits from point of salinity stress, from point of water and nutrient availability, root and shoot related phenotype or phenotyping should be considered, should be taken into account. Usually crop traits for selection of crop traits we use shoots parameter but not roots. But we argue that root is as important as shoot. Next, this is all together maize, sorghum, and alfalfa as a root biomass as a function of collar diameter. We can find that parameter of the power function is really could be related to root biomass, root to shoot ratio, also change of root biomass depending on soil condition. Suggestions that various effect of abiotic stresses will be right in the line of this curve. Then it can reflect contribution of these curves, these relations can reflect contribution of water and salinity or nutrient stresses. So it means that using this line, this is this relationship, we can figure out what could be our root biomass depending of ground stresses. This is how root biomass affects soil hydraulic, biophysical properties. I will not concentrate too much on that. Depending of root rise deposit, not root side but root deposit relationship between soil aggregate stability and soil organic matter. There were linear relation between them, but this relation not directly related to root biomass rather than root resid deposit or soil organic carbon associated with root types or root chemical composition, but it was not our object to concentrate here. We just show that both root biomass and root deposit is important for that, for nutrient, water and salinity stress. Here, our conclusion sort of I was very fast that allometric relationship can be applied for the field crop. Before it was applied just for wood species, for trees, but now we have applied it for plant root. It is difficult, but we have sometimes to do it in order to go next step. The effect of field crop genotype on root biomass is allocable under various environmental conditions. This study provides new allometric model for predicting root biomass from measurements of collar diameter. It is very easy measurement in the field that we can do. The relation suggests that various effects of stress will be light on in the line of the curve. Selection of crop functional traits should be based on both root and shoot related phenotypes. Allometric model can reflect water and nutrient or salinity. Stress result can assist with selecting functional traits that maximize water and nutrient use, and carbon sequestration should be and could be linked to climate change and sustainable or conservation agriculture. Thank you.
Thank you, Doctor.
Yes.
We'll open the floor for if any question or from colleagues online. Okay, I don't see any question, but I have a question for you, Professor or Doctor.
Yes,
in terms of this allometric model to predict root biomass, that's my understanding. I mean,
yes.
What will be the final use? Is this prediction will be at early stage of the crop or at a late stage? And you said in your conclusion that could help in identifying threats for stresses, but could also be this linked to recommendations to in terms of water application, in terms of nutrient application, if the model could be applied at early stage of the crop?
This collection of samples was done at the end of vegetation. It's in harvesting stage. You are right, not continuously. At the harvesting stage to figure out to the harvesting stage what will be common maximum root biomass for next year, how much carbon we will leave in the soil, and what kind of root biomass, what amount of root biomass we will keep in the soil. And plus, on the base of root biomass, we try to figure out what will be contribution of by analyzing root biomass and the end of vegetation. By now, soil properties, what will be contribution of soil properties? This is just examples that I have shown now.
Okay, thank you, Professor.
Yeah.
I guess we'll move to our last presentation. So if you could share your presentation, Doctor. So the next presentation is on the effect of salinity stress on corn during seedling stage, given by Parajakta Pautikar and his collaborator. I hope I pronounced your name correctly. So if you are online, I'll give you the floor.
Yes, sir. Yes.
And if you could just present yourself a little bit for the audience so they know more about you. So the floor is yours. You can share your presentation and start.
Yes, sir. I will share my presentation.
Still waiting for the presentation?
So there is trouble for presenting.
You have problem of sharing your presentation?
Yes, sir.
Try again, we'll give you some time, no problem. Don't be stressed. Take your time. Huh? Yeah, on a team, there is an arrow on the top of the screen that says share. So you just click on that, and if your presentation is open on your computer, you just click on it and it will be shown.
I'm presenting on my phone.
It seems like you have a problem of sharing. Can you just talk? Or
Yes, sir.
if you could send it to us quickly, we can share it for you.
Okay, sir. I'll send you.
Okay. Which name you are going to use for the email address? Because there are too many people here.
It's my email ID.
Huh?
Sir, my email ID.
No, can you give us your email address? Can you give her your email address? You will receive an email address now and you share on that.
Sure, sir.
But you could wait for this, no? No problem. I'm just asking the audience if they can wait. Because the that's the last one for this session. لا دابا هذه آخر وحدة.
Sir,
البريك كوفي.
I send my email ID. I send my email ID on chat box.
You send it to?
It's on chat box.
Can you?
You need to send your presentation to us. Did you understand?
Yes.
Let me write my email ID and you send in this one.
What, she can see this?
She cannot see this one, huh?
Okay, sir.
Krishna, you wanna?
Yeah,
This is
maybe
French.
you can give your email ID. Yeah, or anyone.
Okay.
Yeah.
Can you see the email address on the chat?
Yes, sir.
So we can
I'll
use-
send it.
Okay. Okay. Sorry. It's incorrect. There's a instead of P, there's an S. I will share it. I misspelled my name.
Okay, sir, no problem.
Okay. So this one. Okay, so how can I see this? Did you send it? عندك؟ وباقي ما وصلتليش. ها؟ Yeah, I guess with the time constraint.
Yes, sir, give me 1 minute.
You did? I think I will. Yeah. Well, I guess I'm in the obligation to just stop. We can reschedule that presentation. Just share it with us. Because of the time constraint, the session will continue. Other colleagues are waiting. I apologize and I thank you for being with us without sharing, but it's okay. Hope that for the next time you will be more ready for these kind of things. So thank you very much, and thanks for everybody. I guess this part of this session is ended. We have some times for a coffee break, and then we come back for the second part of the session. Thank you.
شوفوا ترى يشبه دمغت ولا شنو طاحت على راسها لا من قدام يشبه
Hello, good morning, everyone. Could we start? Yeah, with I think our speaker will be Kamal Abrakani, yeah, with the presentation, effect of water stress on leaf photosynthesis and yield of melon and tomato crops grown under Mediterranean condition of the North Sea, northeast of Morocco. The floor is yours, Professor Aberkani. Yeah, you can use this.
So, ladies and gentlemen, it's a pleasure today to present my work on irrigation. Actually, we use the effect. Could you please? Yeah. Yes, so the effect of water stress on leaf photosynthesis and yield of melon and tomato crops grown under Mediterranean condition on the northeast of Morocco. This is part of a PRIMA project that we have with 5 partners from Europe. So we are going to test, I'm going to present some results of this project. Okay. So as you know, the United Nations Climate Change Conference held recently in Glasgow adopted an agreement to accelerate process in order to face the global warming. So as you know, Morocco is among the African countries affected by irregularity and the decrease in rainfall in the last decades, and especially this year. So agriculture, which is the most important economic sector in the country, will have to deal with this problem of water scarcity for the next years. So recently, the focus of research on water management is an important step in order to decrease the impact of drought on field production. And when we are talking about drought stress, we talk also about the problem of salinity, of course. So many research studied the impact of water scarcity on physiological aspects, such as plant growth, photosynthesis. So we can find a lot of research in this, in this topic, of course. So, I'm not going to go more in details for, the literature, but just to say that the objective is to test in the field in the real commercial scale. Because when we grow plants in the greenhouse or something like that, sometimes the growers, they say that it's not the real condition, right? The yield in the lab is not the same thing as the yield in the field. So my experiments here that I'm going to present is more about large commercial scale, about 20 hectares of melon in area of the northeast of Morocco, in Drought and in Nador. And also what we are testing, because Professor Jose Luis Aros, he presents something about stomatal conductance, and it's like the index of stress, but I can say also, the light, the transfer between the electron between photosystem 2 and 1 during the photosynthesis, the chain is interrupted when we have the stress, or we talk about photoinhibition. So I am talking here about these ratios FV, F0. We find those in publication, but there is also other parameters that I use in these experiments. We have around 60 parameters that we can use. I will give more details after. So, the objective of this research work is to study under the Mediterranean climate condition and at commercial production level the impact of different drought strategies of irrigation on chlorophyll A fluorescence or stress index, and the yield of melon and tomato, which present one of the main crop productivity for the country. Okay. So this study was conducted in 2 sites. I think there is a problem. Okay. The study was conducted in 2 sites spaced out at 70 kilometers in Drought in the northeast of Morocco, and the other experiment of tomato in another area. Both sites were realized at the commercial farm production, as I say. So for the melon trial, 2 commercial cultivar we used, Misura and Wifak. Misura is the green melon. Is very famous in this region, and Wifak is the canary, we can say melon, the yellow one. So date of plantation was May 23, 2021. So we make sure that we have the experiment during the hot season. This is for tomato trials. 2 commercial cultivar were used. CV is Karima. They are names, Moroccan names, because they are commercial varieties, and Jed, with plant density of 14,000 plants per hectare. That of plantation was August 28, and the trial was finished on January 25th. So, the tomato trial were a little bit late in fall, not in summer. Okay. So the irrigation treatment were, the irrigation treatment were applied 100%, 75%, and 50%. This is what we made. So we reduced 25% water and 50% of water. So the distance of, because we are not going to close and open, the irrigation, we use the distance between the dripper. So we have 20 centimeters, which is 50%. 15 centimeters between the dripper, it's 75%, and 10 centimeters usually used by the grower. So for the melon, 6 repetition of each treatment were used. Each repetition of treatment contains 6 blocks of 6 plants, etc. I'm not going to go more in details, at least, for the statistics. So I use this, the clip. We dark adapted our leaf for 30 minutes, because we make sure that there is not any transfer of electron during this period, and we give a flash of light, really a flash of light, to saturate our photosystem and to see if we have stress plant or not stress plant, because when we have stress plants, we can say that this chain is interrupted. So the measurements were taken under clear sky condition and very hot condition. At this day, I think it was July, something like that, was like a condition were 42 degrees Celsius in this area. So this is, we made measurements, actually sometimes by myself, by my students. So we go to the field and we measure during all day those fluorescence. So the measurement parameter of chlorophyll A fluorescence indicate the transfer of electron during the light phase of photosynthesis, and they are considered to be good indicator of plant stress. That's as I said before. Okay. So this is the variety of melon that we have. This is the green, is very famous in this area, and the other one we found, which is in the yellow one. Okay. Next, please. Next slide. Yeah, it goes directly to the conclusion. Could you back please to the slide? Okay, next slide after the fruit. No, the next one. Yes, the next one. No, next. Next. Okay, tomato, next. Yes. Okay, here. So actually in this project we are not just trying to reduce the water, but it's because a combined research project with some Italian partners and Portuguese partners. We try also to take some satellite imagery. We try also to have some, this low-cost meteo station, so we can measure the temperature, humidity, etc. of the soil. So we integrate this technology in those trials. So this is the spider model that I show you for the leaf fluorescence. With one light, I can say one flashlight, we can have 60 parameters. Each parameter has a physiological indication. Okay, so in this study, I just take 2 parameters. Those one, they are some data of the humidity of 50% and 100%, so we can have the data online of soil humidity. Okay, I will present some results about the chlorophyll fluorescence because I cannot mention all the 60 parameters that we measure. So you have here the ratio FV/FM. So with with 2 varieties, Missouri and Wifak, we can see there is not significance in melon. Even the temperatures was very high. So no stress. But we can say a tendency here of DI DI is the dissipate heat. More we have plant stress, more the dissipate heat, it's higher, right? So we have, it's not significant, but we can have an increase a little bit in this with water stress. Okay, for tomato, no difference, no significant difference were observed for jad, but for Karima, we can say that here we have the dissipate heat was increased with the stress. So you can have here 185 and 192. So we can see both varieties did not have the same response. Yeah, this is the yield, which is really the most important things for the grower, of course. So you can see, for example, for melon with a reduction of water, you can see Missoura, the yield decreased, right? In 75% and 50%. However, for tomato, even we reduced the water, but not a big significant difference for jad, but for Karima, yes. So we have different response. So as a conclusion, this study shows that the cutting in the amount of irrigation water affected the yield of melon and tomato. Even climate condition of plantation of both crop were not the same. Melon trial in summer and tomato experiment in fall, because we don't have the same condition. Chlorophyll A fluorescence showed different response of each cultivar for irrigation treatments, and the mechanism could be investigated in future study. The project is continuing. Is continuing, and we have other parameters, which is they are satellite, as I said, all these digital, technologies. So thank you very much.
Thank you very much, Professor Aberkani, to be on time. We still have time for some questions. If there are some questions. Yeah. Professor Bolif.
Yes. Oh, yeah. Okay. Thank you very much, Professor Aberkani, for this short but significant experiments. My question is that, the green melon is of recent introduction in Morocco? The green melon,
yeah.
Missoura. Yeah? I've seen it in Spain some 15 years ago or 20 years ago. Is it, how did this variety come to Morocco? Yeah?
I think it's commercial, varieties. Commercial. They call it in this region Drought. Is a very specific place. They grow these varieties. It's Pierre de Sapo. I think they call it Pierre de Sapo. It's a green, it's an hybrid commercials in Morocco. Yeah. So, and this region is specific because the soil is rich in potassium. So the quality of melon in this area is very, famous.
Okay, my question is, did you study the economics of the system under water stress conditions?
No. As you can see in my results, that even some, we reduce in water, right, 75% and 50%, but we reduce the yield. But now we have to see how much it costs the water and how much we lost in yield.
Yeah, it would be interesting to make these economic calculations to
Yes, because I show just only one part of the project. So we have, a big package of the research project.
Thank you very much.
I think she has.
Did you also did some? First of all, thank you for your talk. Okay, then okay. Now you already asked my question a bit, and then I go further with the fruit quality. Did you also check the fruit quality and the texture from the tomato after you reduced the water? If
Yes.
it gets flowery, and if we still want to eat the tomato when you reduce the water. So it's also quite important, even you don't use so much water, but when we don't like the tomato in the end, you just can cook it. Yeah. What about that?
Yeah, we do some quality analysis like pH, bricks, sugar, also the color of the tomato, and we found some differences especially in the color. So we have, I'm not specialist in fruit quality, but I'm trying to do some of the parameter, the dry weight, the fresh weight, etc.
How about the texture inside? Is it more floury when you reduce the water, that the tomato doesn't stay juicy?
No, I didn't.
You don't do
that.
this.
But,
Okay.
but I measure the quantity of
The
water.
quantity of water,
But
yeah.
I found differences between. It's more dried in the dry one. Yes.
Exactly. Yeah. Okay. Thank you very much.
Thank you. Thank you very much, Professor Aberkani. Before moving to the next speaker, I try to call for who is Abu Aqil? Yeah, but we can start for we can move to the next speaker. I think it's Faisal Boghalb. Yeah, our next speaker will be Faisal Boghalb. There will be a presentation on the effect of seasonal environmental change on leaf anatomical response of Lemoniastrum guionianium in Sabha biotype. Are you ready? Faisal Boghalb, Dr. Faisal Boghalb? No. It's not connected. No. So we have to go to the next one. Then the next one will be Mohammed Muwafiq. That will give presentation.
Hello.
Hello. Yeah. Your presentation, it will be drought stress response of 4 contrasting provenance of Argania spinosa. Yeah. The floor is yours, Dr. Mohammed Muwafiq.
Thank you. Thank you.
Please share your presentation.
Okay. Here is clear, right?
Yeah. Could you please put it on the full screen or the presentation mode? Now, at your bottom right, on diaporama.
Backward.
No.
I think I have some issues with the...
No, there is no issues, but in the, yeah, in the bottom of your screen, yeah, close to minus. There is a screen, yeah, that's one. Not yet.
If
you could not, you can go. Okay.
Now you're right.
Okay. Go ahead. Okay.
Thank you. Thank you so much. So it's my big pleasure to be with you here today. I want to thank all the attendees and the participants with us in this conference. So it's my honor to present to you my subject about the drought stress responses of 4 contrasting provenance of Argania spinosa. So as the background for Argania spinosa, the argan tree is an endemic species of southwestern Morocco and the only representative tree of the tropical family of Sapotacea. It's a thermophilic and xerophilic tree with the high atmospheric humidity requirements. It's one of the most forest species in Morocco with area estimated 800,000 hectares, which ranked the third one after after quelques suivers and tetrachloride. The argan tree ensures several functions as ecological function, fight against the desertification, fight against water and the wind erosion. Also has another socio-economic function as wood and fruit, and most popular production, which is the argan oil. The current state of argan tree is in situation of regression under the effects multiple attacks as the climate changes and repeated droughts, overgrazing, extension of agricultural landing, clearing, and domestic over-exploitation. So in this research, we aim to study and characterize some physiological and biochemical traits of argan tree, which is tolerant to the drought stress, and also to determine the oncotypes most tolerant to the drought and able to rehabilitate degraded sites. So as the methodology for plant material, we have used the seedling aged 6 months from the germination of seeds of 4 contrasting provenance, which is Sawira, Agadir, Bizakan, and Berkan. For physiological parameter, we have measured the leaf water potential using the pressure chamber, which call it Schulander, 1965. For biochemical parameter, we quantify the chlorophyll A and V according to the method of McKinney, which most famous, 1941, reading with the optical density in 663 and 644 nanometer. For fluorine content was quantified according to the method of Monovo NMR with the optical density in 528 nanometer. For sugar content, we have used the phenol method of Dubois with the optical density in 485. Finally, for the protein, we have used the method of Bradford, 1976, with the 595 nanometers. So as a result, for leaf water potential, the basic leaf water potential is slightly different between the 2 batches of T1, which is in dark color, and T1, which is in light color, for all the provenance at the start of the experiment. So for Isawera stress ecotype, we found the minimum leaf water potential at the end of the experiment reached a more negative value, which is minus 1.3 megapascal, and the stopping of irrigation. For chlorophyll content, the chlorophyll A and total has undergone a significant decrease in treatment T1, which is the stress plant, during water stress or drought stress. The prevalence of Isawera has reduction rates of 16% in total chlorophyll and 61% in chlorophyll A. For chlorophyll B, we noticed a slight decrease in stress plant compared to the other one, which is the control plant. For the proline content, the highest proline accumulation was noted in stress plant, T1 in light color. For all the provenance, This accumulation rate was 69% for Agadir and 57% for Essaouira. For the sugar, it is the same thing. The highest accumulation was always observed in straight land. This accumulation rate was 8% for Essaouira and 20% for Agadir. It is the same thing for the other provenance. For protein, also the highest total soluble protein content was recorded in T1 plant, which is stress plant, with the rate of
you still have 3 minutes.
Okay, 62% and 30% for Agadir. For principal components of physiological and biochemical parameters, we noticed two groups. Group A, which is contain the provenance of Essaouira and Bizekan with the high protein content. Group B, which contain the ecotypes less tolerant to water stress, Agadir and Bizekan, characterized with high level of proline and total sugar, and more negative leaf water potential. So, as a conclusion, the present study highlighted the interprovenance variation of adaptation mechanism of Arganius spinosa to the drought flores. The four compressing provenance of argan show under the drought condition a significant decrease in water potential as a strategy of water conservation, as well as the significant accumulation of certain osmolytes related to osmoregulation and homeostasis maintenance. These physiological and biochemical responses vary significantly according to the provenance of argan. And thank you for your attention.
Thank you very much, Dr. Mohammed Mouafiq. Thank you to be on time. So for audience, if there are some questions, or if there are some questions on online, you can raise your hand. Yeah, Professor Bolif. Yeah. You can go. Come.
Okay.
It's okay, it's okay.
Yeah, just one comment. I'm happy to hear that our presenter did present the argan tree as a fruit tree.
Yeah.
So 800,000 hectares, while it was a wild tree, considered as a forest species. And I'd like to congratulate the authors for characterizing these biotypes and their drought tolerance. My question was, the presenter started a statement by saying argan is a tolerant, but in the same sentence after, he said that argan was having a great requirement in atmospheric humidity. Could he comment about this, please? Thank you.
Do you understand the question?
Yes, of course, yeah. Argan tree was classified as a tropic forestry tree. So he need the atmosphere to become more humidity, and in the same time, the ability to survive in the highest temperature. This is the biography of argan tree.
Okay, thank you, Dr. Mohammed Mouafiq. Yeah, thank you very much. before going to the next speaker, I would like to ask for our co-chair. I was waiting for him in the room, but I don't know if he's online or not. Imad Abouqila. Professor Imad Abouqila. I don't know if he is online.
Assalamu alaikum. Yes, I am here.
Yeah, sorry. I thought that you will be in presential. I was waiting for you, but till now, yeah.
No, I am here.
Yeah, please. Are you following us?
Yes, I am with you.
Thank you very much. Could you give some notes about the presentation? Do you have the table?
Yeah, in fact, I already now, make the presentation scoring sheet.
Yeah.
And I think it's a very good presentation.
Yeah. Thank you. Yeah. So now we have to go to the next speaker with the presentation title, Using Agronomic Parameters to Rate Quinoa Cultivar Response to Saline Irrigation under Field Condition in Eastern Morocco. That will be given by Ilham Abidi. Did you? Yeah. Okay.
Thank you. Can you share the screen? If not, Thank you. I'm really delighted to be here to participate in such international scientific event, to share with you some outputs of my first trials conducted in eastern region of Morocco, and for the first time as part of doctoral research within IAV Hassan II. My contribution is titled using, please, can you share the, using agronomic parameters to raise quinoa cultivars response to saline irrigation under field condition in eastern Morocco.
Next.
The next. The next. Okay. Because I don't move, you can switch the. So as you know, agricultural productivity is facing serious challenges, and salinity problem is one of them. Continuous soil and water salinization, water scarcity and declining water resources, climate aridity, climate change effects, as well as improper human activity in managing irrigation, fertilization. And here I want to give you some data about global salinization regarding or according to INSEA.com. 1 billion hectares are currently affected by salinization. 60 million hectares are currently damaged by salinization, and 76 million are permanently lost by salinization. Next. Next, please. So, what are the major salinity effects? Salinity reduces staple crop yield. degrades soil fertility and therefore decreases biodiversity, as the majority of staple crops are not able to grow under such limiting condition. And we can see also many of farmers adopting monocropping systems, while others dropping their land towards the nearby towns, seeking a new job to increase their income and to improve their livelihood. All those challenges are constraining the food security achievements, which is the first and the primordial goal of the Agenda 2030 Agenda of Sustainable Development. The next. So, in this condition, how to deal smartly and sustainably with salinity? Maybe introducing salt-tolerant crops such as quinoa could be a sustainable solution to overcome the salinity problems, to enhance salt-affected areas, diversify cropping system, and improve small farmers' income. To do that, we should conduct various research to develop a quinoa technical guide for farmers under saline condition, especially in the east of Morocco, where quinoa is introduced for the first time. Next, please. So as you know, quinoa was introduced in Morocco in 1999 in Khenifra province as part of the project Bafi Bio. Since 2008, quinoa has experienced a new boom and has spread to several regions of Morocco. But in the east of Morocco, quinoa was introduced in 2020 as part of my doctoral research conducted in the province of Barkan and supervised by the IAV Hassan II. And since 2021, quinoa is gaining momentum in the region and got attention for more farmers from other provinces. And now we are in 9 to 10 hectares in our region. The next. Next, please. So why quinoa? Quinoa is drawing an increasing interest worldwide, given its high nutritional value with good amount of minerals, vitamins, dietary fiber, fats, and high quality gluten-free protein with a balanced profile of all amino acids. Its capacity to tolerate environmental contrasts such as frost, drought, and salinity, as it's a facultative halophyte. And another benefit of quinoa, virtues of quinoa, it is socio-economic virtuous by improving rural farmers' income by developing a quinoa value chain. The next. Next. So our specific objective of this, or through our study, is assessing 5 quinoa cultivar productivity under water irrigation salinity levels that are the available water sources, and using agronomic parameters to select the most salt-tolerant quinoa variety. Next, please. So our experience was carried out in a farm located in Barkan province, northeast of Morocco. And our soil and irrigation water characterization, we have a soil with salt loam texture. And our irrigation water, we have 2 sources with 1.5 decisiemens per meter as a control, and 10.5 decisiemens per meters as a saline irrigation. So the climate is the characterization of our climate is a semi-arid climate with an average of total annual rainfall less. Huh? Wow, not yet. Next. So our experimental design is a split plot with 3 replication using 5 quinoa cultivar in the main plot and 2 salinity level of irrigation water in subplot. Our cultivar are Inia 420 Negra, Titicaca, Pinot, Ikba Q5, and Ikba Q4. As I said, we have 2 sources of irrigation with 2 different EC. And the showing and harvesting dates were respectively 13 and March and 10 July 2020. Please the next. So during the cropping period, we have performed some 4 samples to investigate our parameters during cropping grow period. And the parameters that we have measured: height, number of leaves, leaf area, root fresh and dry weight, leaves, and the whole plant dry matter. Next, please. Okay. So our results, according to the table 1, we can conclude that all varieties achieved their growth cycle and developed differently depending on the salinity level of the irrigation's water. Analysis of variance showed a significant effect of the 2 factors as well as their interaction on most of the parameters measured mainly at the flowering and green filling stage. On the contrary, there was no significant effect on leaf area in the last stage. Next, please. Please. So, concerning growth parameters at harvests, the study parameters were total dry matter, grain yield, and kernel, 1,000 kernel weight. Statistical analysis revealed that salinity, variety, and their interaction affect significantly most parameters except 1,000 kernel weight. Please. So, total dry, according to the table 3, we can conclude that total dry matter and grain yield were significantly affected by irrigation water salinity level comparatively to the control, and generally a substantial decrease for both parameters was recorded, although this reduction depended on variety and was more noticeable for Titicaca. You can see in the table. More noticeable for Titicaca with 68%, and Inia, not yet, and Inia with a reduction of 64% in terms of dry matter, and for Pinot and Titicaca in terms of green yield. Moreover, the average yield comparison showed in the figure that the best results were recorded by Ikba Q5 with a minimum loss 21% when it was irrigated by saline water. Please the next. Right? So, as a conclusion, we can conclude that according to this study, the investigated parameters during quinoa growing period were significantly affected by water irrigation salinity at 10.5 decisiemens per meter, which caused the reduction of yield and dry matter at harvest by 66% and 52% respectively. Our funding supports the funding by Razagi, who also reported the reduction in quinoa yield and biomass with increase in salinity level. However, Harish noted that the same cultivar Pinot Titicaca Ikba Q5 and Ikba Q4 cultivated in South Morocco were able to grow successfully above 10.5 dcms, and their grain yield and dry matter were not significantly affected, up to 17 dcms per meter. Nonetheless, our study indicates that this result depends on the quinoa varietal response to salt stress. In fact, Ikba Q5 recorded the highest grain yield with the minimum loss under salt stress, while Inia showed the lowest grain yield under both salinity levels. This Peruvian long life cycle variety should be sown early to avoid high temperature effect, mainly during flowering and grain filling stage. Thus, managing the sowing date is also essential to reduce the negative effect of heat stress during flowering and grain filling stage, and therefore eventual losses of grain yield and biomass. This study should be repeated before confirming these results. And the next, please. No, to tell you thank you for your attention.
Thank you very much, Professor Abidi. So we will have time just for one question. Yeah, two. Yeah, two questions. Yeah.
Thank you very much for your nice presentation. Very interesting. Just this suggestion: is it interesting to add material like organic material, compost, like as another treatment? Because we know that organic material can reduce the effect of the salinity, and also when you have this problem of the stress on your flowering, something like that, is it interesting to add the amino acid, like, or some product? Probably you can reduce the impact of this heat stress.
Thank you, Sia Barkani. Exactly. We have our other essay. When we add, it is the first one. This essay, it is the first one. We wanted to show the response of, first of all, the response of the available varieties that we had to saline irrigation. But we conducted this year in 2021 and 2022 other essays about including other factors like organic amendment, and organic amendment like manure, green manure, etc., and compost. Yeah, thank you.
Okay, briefly, just noticed that Inia gave the highest dry matter, right?
What?
Inia, the Inia variety, gave the highest dry matter, but the lowest grain.
Yeah.
What will happen if you shift the growing cycle from March?
Yeah, exactly.
Will we have the same tendency? Because, I mean, the harvest index is very low.
Yeah.
Thank you.
Yeah, exactly, Dr. Mujahid. The Inia have relatively to our other varieties a long cycle. So we should sow in it before February. So in other essay we can show sown it before.
Thank you.
Thank you.
Thank you very much. Yeah, I'm late. So, yeah, I'm late. We have to go directly to the next speaker. That Kautar Taha. Kautar Taha is here, online. She is connected? No. So we go to the next one. Yusuf Brouzine. Yusuf Brouzine. Dr. Yusuf Brouzine, is he connected? No. Okay. Yeah, we will recuperate time. Okay. Should we go to the next one? So then we go to the presentation of Khadija Mohdi. Khadija Mohdi?
Yes.
Yeah.
Hello, everyone.
Yeah, thank you. The presentation of Khadija Mohdi is titled Effect of Saline Irrigation, Water on Grout and Productivity Grout of Sugar Beet, Beta vulgaris, and the nano irrigation case of moss tube. The floor is yours, Dr. Khadija Mohdi.
Thanks. Okay, I'm going to partage it.
Please share your presentation.
Okay. Is it clear?
Not yet.
Right now?
Not yet until now. It's not yet. Yeah. Put it on the, put it, yeah.
Okay, it's good.
Not yet, we are waiting.
Okay. I put it in a job.
Please, if you can put it on the full screen. Already. Yeah, thank you. Go ahead, please.
Okay.
Okay, thank you.
Thanks. Okay, I can start? Okay, it's good.
Yeah, go ahead, please.
Okay, hello everyone. I hope you are doing well. Thank you for giving me the opportunity to be here with you today. So today's topic is about the effect of saline irrigation water on growth and the productivity of sugar beet under nano irrigation. So studies have shown that more than ever before, the surface of arable land are becoming less distributed, bearing in mind that salt is one of the abiotic stress affecting crops' productivity negatively. In other words, both soil and water as natural resources are poor in terms of their quality, which would also lead to soil infertility, especially in their high salinity level resulting from irrigating with saline water. So in this context, sugar beet could be one of the most suitable options to alleviate salinity stress and boost crop production, since sugar beet is a high salt-tolerant crop with its ability to withstand high water salinity and therefore helps in improving soil health. Sugar beet was chosen also from an economic view as it's considered one of the crops generating more profits. Our objective is to examine the water saving and performance of alternative crop in the nano irrigation. So, which irrigation system would affect of salt stress on the cultivated sugar beet in terms of salt stress? Is it the drip irrigation system or the nano irrigation system? To split my presentation into three main parts. First, I'm going to describe the fluid methodology, and then I will move on to look at the main finding before generating some recommendation. The experiments were carried out during the growing season 2021 at the experimental station of Milk Zahar of Imra in the province of Shtuka Itbaha, which is located for 45 kilometers south of Agadir city. The experiment was laid out in a randomized block design with two factors, the irrigation system as first factor and the salinity stress as second factor, with 2 levels in for each factor and 4 replicates for each treatment, making a total of 11 treatments representing 11 elementary plots of 20 meters square. As you can see, normally we should have 4 replicates for drip irrigation, as we done in the case of nano irrigation, but due to the logistical limitation, only 3 replicates could be realized. However, the unbalanced treatments were taken considered in the statistical analysis. When it comes to data collection, we have measured both soil and plant-based parameters such as organic matter, electric conductivity, chlorophyll content, and root yield. The soil plant samples were collected at flowering stage and at harvest stage. Samples were analyzed at the soil water plants laboratory of the Regional Center of Agronomic Research in Agadir. Using SPS software, we have analyzed the variance among our treatment using ANOVA, and p-value less than 0.05 are considered the threshold of significance. The first figures show the effects of water salinity show the mean value of soil electrical conductivity and organic matter content as a function of irrigation system, salinity treatment, and soil depth at flowering and harvest stage. The results show the effects of water salinity, especially at the beginning of the culture, when we noticed higher salinity in the first 5 centimeters of the soil surface, especially with the drip irrigation system. But at the end of the experiment, it turned out that the difference between the two compared to the combined system, the soil had no salinity effect. Similar organic matter content was reported for the different salinity treatments in the both irrigation system treatment. The studied soil has a buffering capacity, and therefore the flowing uptime is partially short to reveal the difference. When it comes to chlorophyll content, the figure 2 shows the effect of treatment on chlorophyll content at flowering and maturity stage. It was shown to be highly affected with treatments under salinity stress, which means the less chlorophyll we have, the less photosynthesis we will have, and therefore the plant growth process slow down. Then these results are more marked in the nano irrigation condition. Again, the more salinity of water, the more decrease in root biomass we will have, which means our sugar beet can tolerate saline water, but not so much. Acceptable yield of root biomass can be obtained using irrigation water of 6 deciments per meter. However, above this value, we see the significant decrease in yield. The figure 5 shows the change of leaf area in the sugar beet at different development stages as a function of salinity and the applied irrigation system. At maturity stage, sugar beets' leaves were found to be short in terms of their surface area as the salinity level increased. The increased photosynthesis would be related to reduction in leaf area, and the latter has been seen as an adaptation process. Drought improved water use efficiency to encounter salt stress. At the last figure, we show the evolution of proline at different development stages as a function of salinity and the applied irrigation system. It appears that about stage sugar beet leaves respond by accumulating proline to irrigation with saline water, showing an increase in proline content with a p-value less than 0.05. The effects between the two treatments is quite remarkable in the case of nano irrigation. The main results of this experiment suggest that salinity has little influence on the soil physical characteristic in the both irrigation systems. This makes sense because the soil takes a long time to change its property due to its biofilm capacity. The results suggest also that the appropriate placement of the laterals between the crop rows and the dip is well chosen. Then the nano irrigation system would be suitable for the cultivation of sugar beets without decrease in root yield compared to the drip system. So, acceptable yield of root biomass can be obtained using irrigation water of 6 decimals per meter without significant impacts on cultivated soil in the short term. Thank you. Ready for any question?
Thank you very much, Dr. Khadija Mohdi. Thank you also to be on time. So if there are some questions. Yeah, Krishna.
Hello. Yeah. I have a question, Khadija, to you. You compared two irrigation methods, this nano versus drip. Which one method you recommend for the south of southern region, for example, Agadir and down towards the south, considering the profitability or economics? And water application, by the way, you did not say how many millimeter water you applied in those two different irrigation methods. Maybe there might have been some difference in amount of irrigation. So please just consider, I would say three things. One is amount of water applied. Number 2 is yield, crop yield. Number 3 is profitability. Considering 3 parameters, which irrigation method you suggest?
Okay, just a comment.
No, you need to reply.
Okay. Yes. Okay, just for system non-irrigation, it is adequate to the beta sugar beet, okay? But a condition to choose the placement of the lateral, okay? So, but for the drip system, we had remarked that the biomass yield is so important compared to nano irrigation. I don't know if that's clear for you.
You still did not say 2 things. One is how many millimeter water you applied in each irrigation method. Maybe there may be some difference.
Yeah, for the regime irrigation.
Drip, yeah.
Okay, the comparison, you talk about the comparison between the use of water between irrigation drip.
Irrigation method, yeah.
Okay, in this point of economic niveau for water, this part I didn't do it, okay, because the project, it is between us and other partner, okay?
Mm-hmm. Okay.
Thank you. There is another question. Yeah.
Okay.
Yes, Assalamu alaikum, Khadija.
Alaikum salam.
So first, in your presentation, in fact, I am from Egypt. So when I saw the map of Morocco and you mentioned that the site south of Agadir, so I understand that you are from Morocco.
Yes.
But you didn't mention in the first slide about your affiliation or also your other author. So I think you should mention that. Second, the term of nano irrigation, okay? This is my first time that I hear about nano irrigation. So is it acceptable term? So is it okay to say nano irrigation? So we know that nano is prefix, I think it's 10 power minus 9. So how we use the term nano in irrigation? Okay, so this is only my question. Thank you.
Okay, thanks for your question. But if someone can repeat the question, because I didn't understand.
I think it was just a comment.
Okay.
Yes.
Yeah.
Yes.
Okay.
I mean, first you didn't say anything about your work. Where are you working? In your first slide, you just put your name and also other author. Okay.
Yes. Okay.
So yes, I understand that you are from Morocco, from your presentation, but I don't know what is your university and other author.
Yeah.
This is my first, okay. But my question is about nano irrigation. Okay, so I mean, is it acceptable to say nano irrigation, because we know that nano is a term, means 10 power minus 9.
It's
so
it's about terminology.
Yeah.
It's not.
Yes. So nano irrigation is I mean, this term is unique or something new, but is it acceptable to use to say nano irrigation? So I am sure we will use a lot of water in irrigation. So we can't. I think it's nano irrigation is is something strange.
Yeah.
Okay. Thank you very much. I don't. There's another question.
Yes, I have a question. I'm just curious. I know maybe your soils are fertile, but I didn't hear anything about fertilizers used. At least if fertilizers were used, they should be mentioned. I'm just curious.
Yeah, just about the fertilizer that we used in your trial. Do you understand the question, Dr. Khadija?
No.
No.
Sorry, but I don't understand.
About fertilizer that used, les fertilisants que vous avez ajoutés à votre culture. The fertilizer. You didn't mention the fertilizer that you applied to your crop.
Je peux avoir la question en français, s'il vous plaît?
Yeah. Quels sont les fertilisants que vous avez appliqués à votre culture dans ces essais?
Les fertilisants.
C'est pas mentionné.
D'accord. Non, parce qu'en fait, on n'a pas travaillé avec des fertilisants, c'est juste une agriculture biologique, d'accord?
Oui, mais il n'y a pas de fertilisants biologiques de manure.
Non, on n'a pas appliqué des fertilisants là-bas, non.
There is no fertilizer. Yeah, thank you. Thank you very much, Dr. Khadija.
Thank you for you.
So we have to move. We were in advance for time. We have to return back to the presentation of Yusuf Brouzin. Yusuf Brouzin. Do you hear me? No. He is connected?
Yes, I hear you well. Thank you.
His presentation is toward better preparedness of Mediterranean rain-fed agricultural system to future climate change and used water stress. Study case of Bourgreg watershed of Morocco. The floor is yours. I think, it was shared. Your screen is shared. Yeah, go ahead, please.
Fantastic. Thank you. Thank you, the moderator. I would like to start by thanking and congratulating all colleagues, people who are behind this big event. Well, big done. Really big well done here. So my presentation will be about drought, climate change-induced drought, and the impact on crops, mainly rain-fed crops in a watershed in Morocco called Bourgreg. So we all know that Morocco, like many countries in the area, is placed in climate change hotspots where drought is a significant phenomena, and we expect that this phenomena will be a little bit more frequent and probably with a higher extent, higher magnitude, and higher magnitude with higher impact on all systems, food, water, environmental systems. So the point here is to try to find the tool or the, let's say, an approach to understand drought impact on crops, and also to predict this impact in a, let's say, in better way, and also with approaches that require less effort and less calculation efforts. So generally in these tasks we use indices. So there is a big variety of drought and dryness indices that are available that can capture many aspects of the climate and other atmospheric systems, and also the other systems like land, moisture, plants, etc. But one of the drawbacks of this approach, which is used in indices, is the lack of capturing the interaction between more, let's say, more systems, and also their lack of ability in capturing the dynamic feature of these systems. When I mean systems, I mean food, water, and climate. So there are some specialists who think that bringing or using this indices along with modeling, mainly physically or distributed modeling, will help in representing better this dynamicity and also this interference between systems. And this is what we've done. So we try to integrate both drought index and also a hydrologic modeling tool. So the area of interest in this exercise was Grout watershed, which is located in the northwestern part of Morocco. It's a very strategic watershed since it secured a lot, most of water resources for big cities in the area like Rabat and Casablanca. We've got a very strong rain-fed agricultural system there with a wide variety of other activities linked to this activity, which means rain-fed farming in this area has a lot of socioeconomic roles to play in the area. So the objective here, actually it's part of a big project, but I will try to take you through this component of integrating drought index with hydrological or physical modeling. So the objective here is basically how to find a better fit of these indices within the modeling approach, to use this integrated or hybrid approach to understand what's going to be the impact on rain-fed crop yields in the future and the future drought episodes. So here is the approach. I will not take you through all detail, but just to say that we used SWOT models, Soil and Water Assessment Tool model, which is physically based agro-hydrological model with a strong agricultural component. So once SWOT model calibrated, validated over both hydrology and farming components of the Grout watershed, we used the outputs of this calibrated model to generate drought indexes. Here we've got some details about the resolution and also the time step of the used database to set up SWOT model over the area. As a result, here we've got the calibration results of the hydrological components of these models. Since this model is built over a hydrological component, so we need to calibrate first the hydrological part before to go to the crop part. But just to tell you that we've had good results in terms of calibration and validation. That means the model is successful in replicating the hydrological processes in this area. So the calibration was done over 4 different stations to capture most of the water system dynamics in the watershed. Sorry, I've got an interference currently. Okay, I will keep going. Here is the results of the drought index that we picked for this work, which is the SKI, Standardized Precipitation Index, for 12 months. So you can see at your left-hand side the distribution of future drought episodes, monthly drought episodes, over the period 2036 to 2049 under the RCP 4.5. And here we've got the same results over the same time duration, but under this scenario RCP 8.5. So you can see that there are some differences between the two scenarios, which are related to the climatic models that we used, and each scenario has different, let's say, results in terms of what's going to happen in the future in terms of climate parameters. So we used these results to run this the agricultural component of SWOT model to understand what's going to happen in terms of for both water and crop. But let's start by the water component. So in this work we use this indicator, which is total water yield, which is the summary of most of the hydrological components in the area at the outlets of the watershed. So to give a broad idea about water resources over the whole watershed. So you can see here clearly that depending on the scenario, there will be different water budgets for each year. So for example, you can pick from the RCP 4.5, that's where we expect severe drought in 2036, also in 2040 and 2045. So if we come here to see what's gonna be the impact on water resources under the RCP 4.5, which is in green, so you can see that the water resources will follow this trend, and we will have very low water budget in this years, where we will be having drought episodes. And the same for the RCP 8.5, where the very low water budget should be expected in 43, for example, and also in 46, etc. Regarding crops performance, more or less the same trend will be is expected. So under for example, the dryness that is expected in 36, we should have very low yield in the season 36-37. The same for the season of 45, for example, under 45 and 46 under RCP 8.5. So this means that this area is expected to undergo severe drought. This is the first conclusion. This drought will be affecting the water budget of the watershed, as well as the crop performance. There have been a comparison of the drought index that we calculated through SWOT and other in our work with other works. Fortunately, I didn't include it here, but the results showed that the inclusion of hydrological modeling, specifically, and distributed and physically based modeling along standard drought index is helping to capture better the physical processes happening in the area. Thank you. I will be very happy to take any question if any.
Thank you very much, Doctor. So if there are some questions about this topic, online or presential, if there are some online questions. No question? So?
Yeah, maybe I need to, before to close, if you may allow me, I need to mention that this work has been done as part of my previous job at the UM6P Polytechnic University with other colleagues at the International Water Research Institutes and Ibn Zohar University.
Thank you for this information. Thank you very much. So then we have to go to the next presentation. The next presentation, it will be for Leila Radwan but I was just told that unfortunately she passed away, 3 months ago. May Allah have her and or accord her His misericord. So we have to go to a missed presentation for this morning, for the first session. I think it's the presentation of Prajakta Pautekar. Do you hear me?
Yes, sir. Yes, sir.
Yeah, your presentation, effect of salinity, stress on maize, maize during seedling stage.
Yes, sir.
Could you share your presentation? Okay. Please, if you can put it in the, Okay, thank you. Go ahead, the floor is yours.
Okay, sir. Thank you, sir. Good afternoon, everyone. Thank you for giving us this opportunity to introduce ourselves. I'm Prajakta Vijay Pautekar, and my project partner is Ankita Uday Tiwari. We are going to represent our project topic on effect of salinity stress on maize during seedling stage. When it comes to farming, what problems farmers facing previously is very important. So I will go towards the introductory part. So this project is based on the particular crop that is maize. Botanical name of maize is Zea maize. Carl Linnaeus was the first scientist who gave these botanical names to all the plants, and the family of maize is Germany, and it earlier researchers that is the Poaceae, and origin of this crop in Mexico, and it is a center of domestication. Maize is a C4 plant. It means the formation of 4 carbon compound, that is carbon fixation pathway. Maize is a day neutral plant, and it is highest yield potential among cereals. That's why it called it as a queen of cereals. The nutrient content are present in maize is protein, oil, carbohydrates, starch, fat, food fiber, and energy. There are 7 different species of maize. In India, particularly flint corn is found and also known as the Indian corn. Botanical name of Zea maize is Indurata. Present in yellow and white color. It has the carotenoid pigments for its color. And maize grown in Rabi, Kharif and Zaid season, mostly grown in Kharif season, and also used as a Rabi crop. Optimum temperature required for germination is 21 to 22 degrees Celsius, and for overall growth is 32 degrees Celsius temperature required. And this crop is good in sources of vitamin A, B, and E. Indian ICR was the first organization who introduced All India Coordinated Research Project. In 1957, on maize crop was successfully done in 1961. First hybrid variety is Ganga 1. So, the next factor is salinity stress. If high concentration of Na plus NaCl, we refer it as a solicity. And a high concentration of total salt like calcium, magnesium, etc. It referred as a salinity. Salinity depresses growth and photosynthesis in sensitive species. Stress is usually defined as external factor which adversely affects plant growth, metabolism, development, and productivity. There are two stresses, biotic stresses and abiotic stresses. Plant will grow where salt concentration is high. In this condition, salinity stress are high in plants. Our next subtopic is review of literature. Many scientists studied on this topic like drought, salinity, biotic factors, and maize cultivation, and so on. Next is the objectives. Our first objective is to determine physiological effect of salinity stress. to maize by using various concentration of NaCl. And second objective is to evaluate morphology characteristics of maize for salinity stress by using concentration. The study of morphology, homogeneity, size, shape, their color, shoot length, and particular duration of plantlets. And third objective will explained by Ankita. My project part.
Good afternoon, everyone. My name is Ankita Tiwari, and I'm his co-partner. So our third objective is to evaluate biochemical characterization of maize by using different biochemical tests by using various concentrations of NaCl. Here we use different concentrations like 0 millimolar, 50 millimolar, 100 millimolar, 150 millimolar, 200 millimolar, and 250 millimolar. Basically, concentrations are used for observing the plant genetic growth and plant structure. So our next topic is materials and methodology. So here, the seeds were collected from Nat Seeds, India. We have collected 6 different varieties of seeds like Hybrid 5402, Dominator 401, Sona 5101, Dawn 1588, NMH 1707, and NWMH 2002. We were sown it in the germinating tray, and we were used cocoa as a media. After the 7 days of sowing, we observed the characteristics such as germination percentage, shoot length, and after 14 days, we observed variations in the germination percentage and shoot length, and we checked out the root length after 14 days. And again after 14 days, we will extract the chlorophyll by Ornon methodology. So, next slide is our result, next topic, and explained by my partner, Prajakta.
We don't hear you.
Hello.
Hello.
Yeah.
Hello.
You finished your presentation?
No. Prajakta explained the germination percentage.
Yes, sir. So here is germination percentage of our maize crop. Uptake software used for calculating the mean. We use this software to calculate our mean of. Hello, can you hear me?
Yeah, we hear you. Go ahead, please. We hear you.
Okay, so okay. There are 6 varieties we use. First is Hybrid 5402, second is Dominator 401, Sona 5101, Dawn 1588, and NMH and NWMH. In this graph, we observe the germination rate of 7 different varieties after 7 days of sowing, and Hybrid 5402 shows the high germination percentage at 100 millimolar, and variety NWMS shows less germination percentage at 200 millimolar, and remaining varieties are intermediate. Next, shoot length is explained by Ankita.
You still have 3 minutes.
Okay, sir. Shoot length. The next one is shoot length. After 7 and 14 days, variations was observed in the shoot length of all maize treated with NaCl solutions, soil straits. Then we observed this all, and after we calculating the table and graph, we observed that the variety Dominator 401 shows high shoot length at 150 millimolar concentration, and variety NWMS 2002 shows lesser shoot length at 100 millimolar concentration, and remaining varieties show intermediate characters. Then after 14 days, our next is our varieties, effect of salinity on maize crop. So our first varieties like hybrid 5402, then Dawn 1588, NMH 1707, and WMH 2002. And next one is root length. After 14 phase, we observe the root length of maize varieties. Observe those were treated with NaCl concentration. So the variety Dawn 1588 shows higher growth of root length at 150 millimolar concentration, and Dominator 401 shows the less growth of root length at 200 millimolar. Then next one is chlorophyll content. So our main aim is to calculate the chlorophyll content. So chlorophyll is an important photosynthetic pigment to the plant, largely determining photosynthetic capacity and hence plant growth. In the studies, the leaves of 324 plants were collected from 6 different varieties of maize crop. We measured total chlorophyll content. The result shows that the hybrid 5402 shows high chlorophyll content at 645 nanometer, and the variety Sona 5101 shows less chlorophyll content. Then this is the graph of chlorophyll content, and this is our chlorophyll concentrations. So next one is the conclusion and significance. From the observation of morphological characters, concluded that the maize plant treated as a control shows normal growth and shoot and root. The plants treated with 250 millimolar shows less reduction in length of shoot ends compared to the control growth. There were positive results obtained from the chlorophyll content. These are our references. And okay, thank you, sir.
Thank you.
Thank you very much for your presentation. If there are some questions, we open for some question online or presential question. There is no question. Thank you very much for your presentation and your participation.
Okay, thank you, sir.
Yeah, thank you.
Thank you, sir.
Thank you very much for your presentation. So I think it was the latest presentation of this session. So we close this session, and thank you for your presence and your implementation. Thank you very much.
سي محمود
وي أستاذ
أستاذ بخير لاباس الله يبارك الحمد لله. Apparemment, j'ai fait une petite recherche. J'ai trouvé que vous avez la présentation pour la salle, la session B. Vous avez attaqué la session A.
أنا في لا سيسيون
Ah, tu as cliqué apparemment sur le lien de la session A.
Ok, donc je quitte pour aller ailleurs, ياك
احنا نورمالمون انا عندي واحد une autre présentation كنقول كيف بيزار j'ai fait une petite recherche لقيت هاد الشخص كاين cliqué sur le lien ديال la session A au lieu ديال la session B.
لا سيسيون بي اللهم يسر يلا الله يعاونك
désolé مرحبا
مرحبا
سي خالد السلام عليكم.
وعليكم السلام اهلا وسهلا.
كيف لاباس عليك بخير؟
الله يبارك
اليوم
فيك.
بخير؟
صافا الحمد لله شكرا لله بارك الله فيك.
va bien الخدمة؟
الحمد لله لا recherche ça avance.
الحمد لله.
كاينه شي حاجه ومازال كنقلبوا على الباقي.
Oui, ça c'est sûr. Ça c'est sûr.
Donc le programme va apparemment dans le bon sens. Il n'y a pas de retard, il n'y a pas de...
Oui, apparemment c'est bon. Oui. Apparemment c'est bon.
D'abord c'est 14h20, en principe on doit commencer d'abord.
Justement.
Alors juste pour donc c'est 10 minutes de présentation et 5 minutes de discussion.
Exactement, exactement.
Il y avait des changements.
S'il y a des retards ou quelque chose comme ça, on va essayer de réduire la partie, les 5 minutes des questions. Comme ça, on sera... Bon.
Très bien. Pendant les participants.
Donc le scoring sheet, comment on va...
Alors, je n'ai aucune idée là, de scoring sheet. Je viens de l'apprendre.
Ben juste pour évaluer les présentations, pour donner une note.
Oui. Ah oui, oui.
C'est le fichier Word qu'ils ont déjà envoyé.
D'accord. Maintenant, la session de nous, on a 4 présentations.
Je crois 8.
8. Ah oui, c'est une session qui est à 4 et l'autre qui est à 4.
Exactement, oui. Exactement. Oui, tout à fait.
Tu peux partager avec moi le scoring sheet? Je ne l'ai pas reçu, franchement, je ne l'ai pas reçu. Je fais une petite recherche sur la boîte. Ça peut-être la conversation.
Ok. De toute façon, si vous ne l'avez pas, je peux le remplir.
Hello, c'est Khalid Mohamed.
Hello.
Hello.
Yes, we are all of yours.
So just please bear with us. We will resume at 2:40, inshallah.
Okay.
Another 20 minutes, please. Because we have physical participants here, and they still eating their lunch.
Okay. Hopefully you have physical participant, not chemical participant.
No, physical. You are the chemical participant. Thank you, Khalid. See you then in 20 minutes.
Okay, okay, thank you.
I'm trying to send the sheet, but I don't know if it's possible through the chat section.
Okay, then you can send it by email. I will send you right now my email.
Okay.
تعمر إذا بغيت تعمر أنت ديريكتومون لي فورميلير غير هذه جوست بوغ avoir une idée على
Ouais, d'accord. D'accord. C'est bon, c'est envoyé.
Hello guys, in session A, I think Azim, are you there?
Yes, I'm here.
Very good. So, Ahmed from the IT, from the La Régie, are you ready?
Test.
I think
Test.
Khalid, I think you can start the session by presenting the first presenter.
Just to be sure about the first presenter, normally we should start at 20 past 2 with Shayma Afi. And now we have another presenter, and I think
Into the program, so we're gonna start by Shayma. Are you ready? You can share. You can start sharing your presentation, please. Start.
Excuse me. Excuse me. My presentation should be at 2 and 30.
Yes,
My name is Ahmed Fatimi.
Yes, just to be sure about the program, because we had a 20-minute delay, we should start at 2:20, and now we have 20 minutes of delay due to the lunch break. So we were going to start with the first one, which is Ms. Safi, and we're going to go by the role, by the normal role. So we have a delay, and we have to assume it. Just to be a little bit ready for the next coffee break. Okay.
Okay. Thank you so much.
So hello everyone. I'm very happy to be with you, even with the hybrid modes online. But unfortunately, I couldn't share your lunch. But maybe next time. And I hope that you had a great lunch and you have refreshed yourself to follow this parallel session. I think it's the technical session number 1 is very long. It lasts for the first day and the second day alone, and I think it's very rich in information and very rich in new scientific evidence that we're going to discover. And in my list, we have Ms. Shayma Afi, who's going to start with the session with the presentation titled Saline Water Irrigation Effect on Yield and Quality Oil of Algae Trees. Shayma, are you ready?
Yes, I'm ready.
Okay, you have 10 minutes, and we are going to discuss a little bit about your presentation, about 2 or 3 minutes, just to be short, to allow your colleagues to present their work also. Okay? You have the floor. Thank you.
Can you see the screen?
Yes. Yes. Turn it in full screen.
Yes, I'm going to put it in. Can you see the full screen now or not?
yes, go ahead. Timer is on.
Okay. So my name is Shayma Afi, and I'm a second-year PhD student at the University of Ibn Zuhra, Faculty of Science, Laboratory of Biotechnology and Valorization of Natural Resources. And at the same time, I'm an intern at the National Institute of Agricultural Research. So today I will be presenting to you my work, which is entitled Saline Water Irrigation Effect on Yield and Oil Quality of Algae Trees, domesticated in Laayoune, Morocco.
But before I start, I would like to thank you for giving me the opportunity to participate in this event. I would like to thank the organizing and scientific committees for their effort. Are you still able to see the screen?
No.
I think somebody else shared their own, I'm not sure.
Share again, please share again.
Okay. Just a minute. Is it on?
Yes, go ahead.
Okay. As I said, I would like to also thank the organizing and scientific committee for their effort, and I would like to thank my supervisors, Dr. Hilam Jamal, Dr. Mimonia Al-Khatif, Dr. Naima Aitabid, and Professor Muad Mshanda for their constant support, availability, and encouragement. So without any further delay, we all know that there has been an increase in demand for water, and with the climate change shift, this only seems destined to aggravate. Therefore, agriculture, in order to respond to the growing demands of the population, will be with time obligated to use water with low quality, such as brackish water or recurring effluent. And the use of those type of waters will unfortunately increase the risk of soil salinization or a reduction in yield. And, in Morocco, which is a country where we have all the variants of the Mediterranean climate, we have the phenomenon of drought, which has led to a process of soil salinization. So these two natural constraints are in fact the main cause or the reason behind the soil desertification. So among the different strategies try to overcome salinity, there is a technique of drainage of salt excess. Yet due to the fact that it's not affordable, the introduction of plant species to tolerant to salt stress of a great socio-economic value is one of the possible approaches to rehabilitate soils. Also, we have our famous Zinc tree, which is known to be an endemic species that covers approximately 870,000 hectares. It's known also for being able to grow along the coast between Safi and Ifni in the South Plain and on the flanks of the western parts of the height in the entire atlas. It's also known for its characteristics such as being xerophilic and thermophilic, meaning that it can develop in the semi-arid and arid temperate and even hot conditions, as well as the relative humidity in the air is sufficient. So Zinc tree is known to be a tree of a great social economic interest, that it produces an oil much sought after for its nutritional and cosmetic qualities. And all of those virtues put the Zinc tree in the course of the problem as much as a tree or species tolerant to salinity. Our study, moving on to the methodology that we followed, our study took place in an alpine orchard that, in the experimental field in Laayoune city. This experimental field, in fact, belongs to the National Institute of Agricultural Research, and 12 Zinc trees that were planted in 1996, and it has a soil that is characterized by a sandy, loamy, and clayey texture on a limestone. So our trees were irrigated with approximately 40 liters of saline water twice a month, and the water was characterized by an electrical conductivity of 16.45 deciments per meter and a thorium content of 80 ppm. I must also state that during the time of the study, only 8 of those trees were actually productive at a time. In order to present or to prepare our plant material, we proceeded by a pulping to remove the pulp, a crushing to remove the nutshell, grinding to obtain the powder-like format, and a drying of that powder at a 40 degrees C for 24 hours in order to remove the excess humidity or water. And to extract our oils, we used the ultrasound-assisted extraction. It is a technique that uses ultrasound to generate rapid movement of solvent, resulting in a higher mass transfer speed as well as an accelerated extraction. So we took 10 grams of our powder with 80 milliliters of solvent. We use hexane in our case. And then we put it in the ultrasound bath for 45 minutes, 50 degrees, and at frequency of 28 kHz. We filtrated this using the filtering paper, and we ornamented the solvent using the rotavator to recuperate our oil. So our oil, in fact, at that time, we took some measurements to calculate the oil yield. And to study the quality of the oil, we studied these different parameters, such as the total phenolic and flavonoid content, antioxidant activity, and we determined our fatty acid profile. To do so, we prepared our methanol extract using the method proposed or given by Property 8 Middle. For our total phenolic content, we determined them using the protocol described by Rebay. So the principle is at the oxidation of phenols, a reduction in the following clear-cut reagent occurs, which results in a change of that reagent from a yellow to a blue color. As for the flavonoids, we determined them using the Bogandora and the protocol. We use the aluminum trichloride and sodium nitrate, which gives a yellow complex or a pinkish type of a colored complex when the flavonoids are present. Activity, we use the DPPH method, which is a free radical of a violet color that changes to a yellow one when neutralized by antioxidant. Fatty acid composition was determined using gas chromatography. To study our analysis, our data, we use statistical software version 10. So now
we still have 3 minutes.
3 minutes?
Yes.
Okay. So for our oil yield, we observed that for the oil yield, we remarked that our yield varied between 27.04 and observed in the C4 and a maximum yield of a 47.60% observed in the C1. So the yield that we obtained were in fact lower than those mentioned in previous studies and literature who reported an oil yield with solvent ranging from 50 to 57%. So we thought that this could be explained by the fact that these yields were obtained with different extraction methods. And we also thought that maybe this could be explained by the fact that it's linked to a genotype effect. As phenol content, total polyphenol content. So we observed that we have a variation of content that varies between 0.075 milligram equivalent of gallic acid observed in the A tube and 0.093 observed in the C tube. So the content in all our oils surpassed the content obtained by a previous study who found a value of 0.072 milligram equivalent of gallic acid per gram of oil. And it was an oil, in fact, that it was extracted using a mechanical press and originally from Israel. So we thought that the difference could be explained by silent stress, since polyphenols are a secondary metabolite produced when the tree is at a stress phase. Flavonoids content, we found out that it varied between 1.749 milligram equivalent quercetin in C4 to a 2.494 in C1. When we express
1 minute, you still have 1 minute.
Okay, I'm going to try to maximize. Okay. Our values were surpassed by those found by another study who found a 1.77 milligram. Antioxidant activity, we found that our values varied between 22.80 and 46.17. Those results were surpassed, in fact, by other studies that found 80% or more. Composition were all during the range of the Moroccan standard, and analysis of variance proved that for the oil yield we have no significant effect on salinity. There was a high significant effect on the polyphenols and flavonoid content as well as for the palmitic and arachidic acid. To sum up, our results suggest that except for the secondary metabolites, all parameters are probably either genetically controlled by the tree trees or are affected by biotic or abiotic factors such as type, soil type, climate, age, and extraction method. The water salinity remains an inevitable challenge, and despite the effects of salinity on the major byproduct, which is the iron oil, this technique, this unique in fact tree represents one of the best choices in tolerance and adaptation to climate change.
Thank you. Thank you very much for your nice presentation. Actually, we learned a lot about algae trees and the effect of saline water or salinity on this endemic tree in Morocco. So maybe one or two we can have 2 or 3 questions during this 3 minutes we have to discussion. And the floor is yours. Who want to ask a question? Hushaim, of course. Okay, Shaima, your presentation was very clear, so no questions are pending. So now we go right away to the next presentation, titled The Emission-Based Formulation for Increasing the Resistance of Plants to Salinity Stress, a Patent Evaluation. That's interesting, a patent evaluation. From our colleague Ahmed Fatimi. Ahmed Fatimi, are you ready?
Yes, thank you, dear Azim.
Okay, you have 10 minutes and no longer than 10 minutes, please, okay?
Okay, thank you.
Go ahead.
That's okay, you can see my full screen?
Yes, you can see the screen, but just turn into full screen. Great,
It's okay?
Go ahead.
Okay, good, thank you, dear Azim. First of all, I would like to thank the conference chairs, Professor Harish and Professor Shokrallah, and the reviewers of this conference for giving me the opportunity to present this work. So this work has been carried out by my postdoc at Sultan Moulay Slimane University, Dr. Fatima Zahra Hafian. So, as you know, the protection of plants from environmental stresses is one of the major concerns in the field of agriculture. In agriculture, conventional plant treatments are generally enabled to provide plants with resistance to abiotic stresses and are therefore limited to providing benefits to otherwise healthy or flourishing plants. However, commercial agronomical processes require additional plant treatment to reduce plant stress or enhance the plant's ability to resist common abiotic stresses.
Ahmed, we can't hear you. Please unmute.
Ahmed, unmute. Your mic is muted. Look, unmute your mic from MS Teams, not from the laptop. Can you hear us, Ahmed?
Okay, that's okay now? It's okay?
Yes, that's okay.
Okay. So a typical example of common abiotic stress in marginal...
Full screen, please. Full screen, please.
A typical example of common abiotic stress in marginal lands and sea basin includes continuous exposure to saline water, which cause swell salinity. So salinity stress results in poor or no yield, mainly due to reduction root grout, as well as reduction the nutritional status of plants and or fruits. More specifically, salinity directly affects the morphology, physiology, and metabolism of plants. So this work in the form of a patent evaluation describes the use of an emulsion-based formulation to increase the resistance of plants to salinity. It should be noted that patent evaluation is a technique for studying the information present within and attached to patents, However, it describes the states of the art by introducing what has been patented in relation to formulation for increasing the resistance of plants to salinity stress, both regarding preparation methods and processes and application. So the international patent classification of this patent, patent number United States 2016-03024116E1, concern generally preservation of plants, biocides, past repellents, attractants, or plant ground regulators containing hydrocarbon and or heterocyclic compounds and or mineral oils. So throughout this patent, inventors discovered and claimed a method and an emulsion-based formulation for increasing the resistance of a plant to damage caused by one or more abiotic stresses, among others, salinity stress, which comprises applying an emulsion amount to the plant. The claimed emulsion-based formulation comprises paraffinic oil, emulsifier, pigment, water, and additive. So you can see here in the picture the reference of the evaluated patents used in this study. So a typical effective agricultural combination of this oil in water emulsions is presented in the table. So as emulsion components, you have paraffinic oils, emulsifier, pigment, water, and additive. Two studies concerning salt stresses were carried out to prove the concept of this inversion. Actually, oil in water emulsions compositions were tested. So we have composition A, composition B, and composition C. So the two studies were initiated in the greenhouse to look at the impact of the three above cited compositions, A, B, and C, on spring wheat and turf grass grown under salt stress conditions. So the first salt studies were conducted on spring wheat in the greenhouse. Wheat seeds were grown in the greenhouse under full light for 2 to 3 weeks to react to the tree leaf stage, and the wheat plants were treated with the compositions E and V by foliar application. The treatments were performed on different samples, samples 1, 2, 3, 4, and 5, and you can see in the table the different condition as a function of salt treatment and the composition A and B. The second salt stress studies were conducted on turf grass grown in the greenhouse. Turf grass seed was sown in potting soil in the greenhouse under the full light approximately 4 weeks before the trial start dates. Upon trial initiation, pots were watered with different NaCl concentrations, and the treatment of at different concentrations were performed on different samples under the conditions presented in the throughout the table number 4. As results for the first salt stresses study, each treatment showed no significant difference in the plant height. However, the measured biomass for each treatment by fresh whited and dry whited showed significant difference. So all these results, it's already presented in the patent referenced in the title. The treated plant under salt stress also exhibited similar biomass as an untreated control, which was not subjected to salt stresses.
We still have 3 minutes.
Okay, thank you. As a result, you can see here the results as a function of different concentrations of NaCl, and we can assume that the turf grass showed no significant scene of the stress during the first two applications, 0 and 0.03 molar, and no significant difference between the treatment. Turf quality ratings highlight results.
Please, can you share, Ahmed, can you reshare your presentation because we lost it?
Okay, sorry. It's okay now?
It's okay.
It's okay? Okay. So turf quality ratings highlight results assessed 35 days after initial Fourier applications or 49 days after initial Fourier applications, as these were the date when the greatest difference were observed between the composition C-treated and water-treated plants. So as conclusions, soil salinity stresses cause osmotic stress and affect the agronomical, physiological and nutritional status of the plant. The studied patent reference uses an emulsion-based formulation to increase the resistance of plants against salinity stress. Through this patent, the inventors claimed preparation method processes and applications of oil in water emulsions to treat plants under salt stress, and the inventors confirmed that the proposed formulations could increase the tolerance of spring wheat and turf grass against salinity. According to inventors, the method of applying the emulsion-based formulation to the spring wheat or to the turf grass at the onset of the salt stress condition enhanced the tolerance to the stress conditions by improving the yield and the quality of plants as compared to young-treated plants subjected to the same salt stress. In particular, the quality of spring wheat and turf grass was not degraded as quickly or to the same extent as the control. Finally, the inventors proposed that the emulsion-based formulation could be applied prior to or during the crop growing period to improve salinity stress. Thank you so much for your attention.
Thank you, dear Ahmed, Dr. Ahmed Fatimi, for your presentation. That was very concise and 10 minutes. Yes, you have respected the duration of your presentation. Thank you very much. I'm turning now to the audience. Is there anyone who wants to ask one or two questions? Okay, if no one wants to ask a question, I have a question for you. Ahmed, please, can you give us an idea about the economic cost of this patent? Maybe the inventors have already evaluated the economical cost. Maybe you have an idea about it.
So I don't have.
It's a price. It's a cost per hectare. If you can.
So I think I don't have any idea about the commercialization of this product, but regarding to the ingredients to the formulations, it's a basic formulation. So paraffinic oils, it's not too expensive. Pigments, it's not too expensive. Water. So I think it's possible to use this formulation for salinity stresses, but I don't know if the inventors have already commercialized this invention or no. But it's very easy to do. You can do it in the at home. So it's not difficult to prepare this formulation.
I have a question. Thank you. Just for people who want to ask a question, you can raise your hand, the yellow hand, and then I give you the. Okay, go ahead. You can ask. Marwan.
Yes, please. Regarding this product, can we apply it also as a seed treatment or only foliar? What is the method that is being applied?
So we can test.
The second question, please. Yes, this is the first question. The second question is regarding the patent. It's a Moroccan patent or it's American or from another country?
So I have already presented in the preceding paper the origin of this patent. So it's an American patent, but it's also a family of patents. So we have, I think, if I remember, there is the American patent and Canada, United States, Uruguay, and the global system. So we have Canada, United States, and Uruguay. So it's 3 patents inside one family. So for to have more information about this patent, you can consult my proceeding paper in the environmental science proceeding dedicated to this conference. So for your first question, the inventors has already concentrated their research only in foliar, but I think it's possible to generalize this application for another techniques.
Like drip irrigation, for instance.
Yeah.
Or
Okay,
seed treatment.
thank you, Ahmed, for your
seeds come,
presentation.
for example,
Or seed
already
treatment,
in
yes.
Yes.
Okay, no other question? So, great. So let's go to the
If you need just my dear Azim, please, just if you need any help or any information about this study, you can write to my address email. So I will reply you as soon as possible.
Thank you, Ahmed, for your openness.
Thank you so much. Thank you so much.
Now we go to the third presentation by Nasser Asrewa, titled Impact of Severe Salt Stress on Morphological, Physiological, and Biochemical Parameters in Alfalfa. I just want to remind you, the participants, that we don't have to share your screen unless you are a presenter. So please bear in mind that because presenters are not should focus on their presentation, and once you want to use or to try to share your screen, then you don't make the presenter at ease. Go ahead, Nasira, you have 10 minutes. Nasira, can you hear us? Actually, I don't hear you.
Please unmute your mic, yes.
Hello, can you hear me?
Yes, now we can hear you. Go ahead.
Okay.
You have 10 minutes.
Thank you. Bonjour tout le monde, je me présente, je m'appelle Sraour Nassera, je suis doctorante au sein de l'Université Sidi Mohamed Ben Abdullah, faculté polydisciplinaire de Taza. Et je suis très heureuse d'être aujourd'hui parmi vous et de vous présenter une partie de mes études sous titre d'effet du stress salin sur les paramètres morphologiques, physiologiques et biochimiques de la luzerne, Medicago sativa. Alors, comme on sait tous, la salinité est un grand problème qui menace la Terre en entier, puisque 20% des totals du terre cultivé et 33% des terres agricoles sont affectés par une forte salinité. Au Maroc, 500 000 hectares sont affectés par la salinité. Et donc, de trouver des cultures alternatives tolérantes au stress salin peut être la meilleure solution pour exploiter les terres agricoles touchées par la salinité. La luzerne est une plante fourragère très cultivée au Maroc par un pourcentage de 22% du total des cultures fourragères. Le présent travail a été réalisé au sein de la faculté polydisciplinaire de Taza, laboratoire ressources naturelles et environnement, dans une serre sous conditions contrôlées, température de 21 degrés Celsius, 65-15% d'humidité et sous une lumière naturelle et artificielle. Les graines de Medicago sativa sont désinfectées et rincées par de l'eau distillée et mises dans des pots contenant du sable, de la tourbe et des sols. Après 21 jours, le stress salin est appliqué par l'irrigation par de l'eau de robinet 400 microsiemens par centime pour les témoins et de l'eau contenant du NaCl 8 décimètres par mètre pour les stressés. 35 jours après, les échantillons des feuilles et des racines sont stockés à moins 20 degrés Celsius pour les analyser ultérieurement. Les pigments chlorophylliens, le chlorophylle A, le chlorophylle B et le total chlorophylle sont extraits dans le diméthylsulfoxyde, le DMSO, selon le protocole de Burnisson. Les sucres solubles sont déterminés selon la méthode d'EREC. La proline est extraite selon la méthode de Bates. Les protéines sont quantifiées selon la méthode de Laayoune. La matière sèche des échantillons des feuilles et des racines sont séchées 48 heures à 70 degrés Celsius. Alors, comme résultat, on constate que la salinité affecte les paramètres morphologiques de la plante puisqu'on constate une forte diminution de la matière sèche des racines et des feuilles par un pourcentage de 77% et 42% respectivement par rapport aux témoins, et une diminution des pigments photosynthétiques, le chlorophylle A, le chlorophylle B et le total chlorophylle. L'analyse de la variance a montré que les deux facteurs, partie de la plante et traitement, ainsi que l'interaction entre les deux, influencent significativement les paramètres étudiés. Le facteur traitement est la source la plus importante de la variabilité pour les teneurs en proline, en sucre soluble et en protéines. Pour les paramètres biochimiques, la proline, les sucres solubles et les protéines, les résultats montrent une augmentation significative de ces paramètres dans les deux parties de la plante, feuilles et racines, suite au traitement par NaCl. Donc, on constate une augmentation significative de la proline et des protéines dans les deux parties de la plante due au traitement par NaCl. Pour les sucres solubles, on constate qu'il y a augmentation dans les plantes stressées par un pourcentage de 41% et 31% dans les feuilles et les racines respectivement. Alors qu'en conclusion, la matière sèche des feuilles et des racines a été réduite significativement sous stress salin par un pourcentage de 77% et 42% respectivement. Le stress salin réduit les pigments photosynthétiques, la chlorophylle A, la chlorophylle B et le total chlorophylle dans les feuilles de Medicago sativa. Le stress salin augmente les teneurs en protéines, en proline et en sucre soluble dans les deux parties de la plante, feuilles et racines à la fois. La salinité induit une forte accumulation des protéines et des prolines dans les racines par rapport aux feuilles. Comme prospective, il faut donc étudier d'autres paramètres au niveau de la plante et pourquoi pas d'étudier des variétés marocaines. Et merci pour votre attention.
Merci beaucoup, Nassera, pour la présentation.
Merci à vous.
Donc, s'il y a des questions de l'audience, si quelqu'un veut poser une question, il lève la main.
Bonjour.
Oui, c'est qui?
Bonjour, c'est Amina. Juste une petite question concernant le début de la présentation. Madame Nonsseira, je pense, qui présentait tout à l'heure. Nonsseira. Vous avez parlé des paramètres, l'influence de la salinité sur les paramètres physiologiques et biochimiques. On a vu que les paramètres biochimiques. Ça, c'est la physiologie de la plante. On l'a pas vu, à part la masse sèche de la plante. Mais ça, je trouve que ce n'est pas un paramètre physiologique. Ça, c'est de 1. Deuxièmement, la question, c'est comment je demande le mécanisme, comment la plante, sous un stress de salinité, va libérer tel ou tel composé, genre la proline, les sucres libres, et vous avez parlé des protéines. Comment ça? Est-ce que vous faites des études sur le niveau moléculaire, sur le réseau métabolique de la plante, ou juste sur des traits, sur les réponses, genre les réponses biochimiques de la plante? C'est ça ma question et merci.
Juste laissez-moi un petit peu de temps pour traduire. Just for our colleagues who don't understand French, the question was about the parameters presented in the result and discussion section. There's only physiological parameters who have been presented and not the microbiological maybe or biological parameters, and also what is the mechanism involved in the stress tolerance of the plant to the severe salinity or soil salinity, water salinity. This is the question, and now we're waiting for the answer. Nasera, tu peux répondre en français, je vais essayer de traduire ta réponse en anglais.
Merci. Donc, pour la première partie de la question, pour les paramètres biochimiques, les paramètres physiologiques, je pense que la photosynthèse, c'est l'un de ces paramètres. J'ai déjà parlé de la photosynthèse, des paramètres des pigments chlorophylliens, la chlorophylle A et la chlorophylle B et la chlorophylle totale. Pour la deuxième partie de la question, on n'a pas encore découvert comment la plante réagit par la production la mobilisation de ces paramètres. Donc, on est en train d'étudier ça.
D'accord. D'accord, merci.
Merci à vous.
Okay, Nasera just said that more parameters have been presented along with biochemical parameters, and also we don't know how the mechanism have been involved. What is the mechanism involved in the mobilization of all those response to the severe salinity stress? And maybe it's the beginning of PhD or maybe postdoc, and I think that's in every study we have a beginning, and also, of course, we will have too much results at the end. Good luck, Nasira, and if there is no question, we move to the next presentation from our colleague Mohammed Abdullawi. Presentation titled Impact of Water Stress on the Productivity and Quality Parameter of Wheat and their Potential Use in the Breeding Program. Mohammed, are you there? We can't hear you. Maybe you should unmute your mic. Mohammed Abdullawi?
Yes.
Okay. Do you want to start by the conclusion? Is this the right presentation? Mohammed Abdullawi? Hello, Mohammed?
Hello, sir.
If you're not... Okay, Mohammed Abdullawi, are you there?
Yeah, I just have a problem in connection. I will repair now.
Tell me, this is your presentation. Is that okay? Is that right? We're running out of time. Mohammed. Mohammed Abdullawi. Tu peux parler en français, pas de souci. Juste nous dire.
Oui, j'ai un problème de connexion sur PC. Je vais juste le régler pour commencer.
Alors, est-ce que tu auras besoin de plus de temps? Sinon, on peut passer à la présentation suivante et on revient vers ta présentation. Qu'est-ce que tu en penses?
OK.
Now we go straight ahead to the next presentation by our colleague Saïd Bouhrawa. Saïd? Please mute your mic if you're not the presenter. Said, are you there?
Oui, je suis là.
Okay, thank you very much. You have a presentation titled Assessment of Morphological and Physiological Traits of Moroccan Barley Variety submitted to severe salt stress. Do you have a presentation ready?
Oui.
Great. So you have 10 minutes, and we will have about 3 minutes of discussion. Go ahead, timer is on. We share again.
D'accord. Alors, bonjour tout le monde. Je m'appelle Saïd Bourawa. Je suis un doctorant à la faculté de polyspécialité de Zaza. Et je remercie la comité scientifique et la comité d'organisation pour m'avoir donné cette opportunité de présenter devant vous une partie de mon travail. Mon travail intitulé Évaluation des caractères morphologiques, physiologiques et biochimiques des variétés d'orge marocain sous stress salin sévère. Alors, mon travail est réparti en 4 axes principaux. D'abord, on va commencer par une petite introduction. Ensuite, je passerai aux parties méthodologie. Et après, je vais entamer partie résultats. Et je conclure par une petite conclusion. Au Maroc, l'orge occupe la 2e place après le blé en termes de consommation humaine et la 1re place en termes de nutrition des batailles. Comme vous voyez ici, dans cette figure, dans les dernières années, on observe une diminution de la production de l'orge et pour cela l'importation des besoins de ce type de céréales et dépend de la production de chaque année. L'Organisation des Unis pour l'alimentation et l'agriculture estime que 20% des terres irriguées dans le monde sont des terres touchées par le problème de salinité. Comme il est détaillé sur la figure 3, c'est dans les régions arides et semi-arides que le degré de salinité est plus élevé. Alors, il est nécessaire d'adapter l'agriculture biosaline pour améliorer la productivité de ces terres. La recherche des variétés tolérantes pour la salinité pourrait être une solution judicieuse pour surmonter ces problèmes. Alors, comme il est illustré sur cette figure, la salinité a des effets directs sur les différents paramètres tels que les paramètres morphologiques, physiologiques et biochimiques, ainsi que les paramètres de rendement. Parmi ces changements, on note une diminution de la longueur des racines et des pousses, la réduction de la biomasse en général, diminution des pigments photosynthétiques et l'accumulation des osmolytes tels que la proline, les sucres et les protéines chez certaines plantes qui sont tolérantes au stress salin. Dans notre étude, on a travaillé sur 4 variétés d'orge marocain qui sont Messine, la Nasser, Amira et Adrar. L'étude a déroulé dans une serre de la faculté polydisciplinaire de Taza sous des conditions de température et d'éclairage contrôlées. L'expérience est faite par la technique hydroponique. Alors, dans cette expérience, on a appliqué 2 niveaux de stress en ajoutant 0 et 300 millimolaires de NaCl à la solution nutritive. La solution a changé tous les 5 jours. Après 15 jours de l'application de stress salin, les plantes sont récoltées pour les analyses. Alors, pour les paramètres morphologiques, on a pesé le poids sec des racines et des plantes. Pour la chlorophylle, on a utilisé la technique de Bernesson. Pour l'analyse des fluides électrolyques, on a adapté la méthode de Zalé. Et pour la proline, on a travaillé par la méthode de Bates. Ce qui concerne l'analyse statistique, on a fait l'analyse des variances, la comparaison des moyens par logiciel Minitab et la corrélation et l'analyse des composantes principales par l'Excel Stats. Alors maintenant, on passe aux résultats. L'analyse des variances a montré la forte influence du stress salin sur l'ensemble des paramètres étudiés et qui a exprimé plus de 77% de la variance totale. L'analyse montre aussi l'influence de groupes de variétés sur le poids sec des racines et la proline. et l'interaction entre le traitement et le groupe de variétés influence la proline et la chlorophylle totale. Alors, les résultats obtenus pour la comparaison des moyens montrent une diminution significative de poids sec des racines et des pousses chez les variétés sensibles Adrar et Amira par rapport aux variétés tolérantes Massine et la Nasr. La même chose pour les fluides électrolytes. On a une augmentation des fluides électrolytes chez Amira et Adrar par rapport à la Nasr et Massine. Pour ce qui concerne la chlorophylle totale, il y a toujours une diminution hautement significative chez toutes les variétés, mais il y a une diminution plus grande chez Amira et Adrar par rapport à Nasr et Massine. Toujours Amira et Adrar sont deux variétés sensibles par rapport à la Nasr et Massine qui sont des deux variétés tolérantes. Ce qui concerne la teneur en proline, il y a une accumulation élevée chez Massine et la Nasr par rapport aux deux variétés sensibles, Amira et Adrar. Alors, pour la matrice de la corrélation, on observe ici une corrélation hautement significative positive entre le poids total, le poids sec des racines et le poids sec des feuilles. Ainsi, une corrélation positive entre la chlorophylle totale et le poids sec des racines. Il y a une corrélation hautement significative négative entre la proline et la fouille des électrolytes. Cette matrice de corrélation est de contrôle. On passe maintenant aux matrices de corrélation de stress 300 mmol de NaCl. On observe ici une corrélation hautement significative et positive entre le poids sec des racines et le poids sec des pousses, et aussi une corrélation hautement significative négative entre la chlorophylle et les électrolytes, et entre la proline et les électrolytes. Et enfin, une corrélation hautement significative positive entre la proline et la chlorophylle totale. Pour résumer, les relations existent entre les facteurs électriques, morphologiques, physiologiques et biochimiques. Dans notre étude, nous avons réalisé l'analyse de la composante principale. Les résultats issus de cette analyse confirment ce qu'on a trouvé précédemment par la NOVA et par la comparaison des moyens et par la matrice de corrélation. Ici, de PCA, le PCA présente les seuils de contrôle. Il y a une corrélation positive, une association entre la proline et la chlorophylle totale, et une corrélation positive entre les poids secs des racines et les poids secs des pousses, et il y a une corrélation négative entre les fluides électrolytes et la proline et la chlorophylle totale. Alors, Amira et Adrar sont associés dans la partie négative, sont des deux variétés sensibles, alors que Massine et Nasr sont associés dans le côté positif, sont deux variétés tolérantes. On passe à le PC2 dans le cas de stress. Il y a une association.
Il vous reste 2 minutes et demie. Il vous reste 2 minutes et demie.
D'accord, merci. Il y a une association positive entre le poids sec des racines et des pousses. Il y a une corrélation hautement négative entre la proline totale et les électrolytes. Alors, les variétés tolérantes pour conclure, les variétés tolérantes ont démontré une stabilité des paramètres morphologiques, physiologiques et biochimiques dans les conditions de stress salin par rapport à d'autres qui sont sensibles et qui avaient des performances terribles dans les conditions de salinité. Pour les perspectives, on a proposé de studier d'autres paramètres tels que les paramètres moléculaires et ainsi de suite, et de tester plusieurs variétés telles que les variétés des pays méditerranéens. Alors, merci pour votre attention.
Merci beaucoup, c'est Saïd.
Merci.
Alors, s'il y a des questions, vous pouvez lever la main ou bien poser la question directement. Je m'occuperai de la traduction. Is there any question?
J'ai une question pour Saïd.
Oui, vous vous présentez?
C'est professeur Mohamed Aït Mokhtar.
Ah, voilà. Vas-y.
Vous avez étudié l'effet de la salinité sur les 4 variétés. Sur les paramètres, les paramètres que vous avez signalés, est-ce que vous n'avez pas essayé de voir, évaluer l'effet de la salinité chez ces 4 variétés, mais concernant le rendement? Parce que ce sera très important de savoir quelle est la variété la plus tolérante et qui donne plus de rendement.
Oui, alors dans ce travail, juste une partie de mon travail. Si je vais présenter tout mon travail, il y a la contrainte de temps et et plusieurs contraintes. Moi, j'ai essayé d'autres variétés, les variétés méditerranéennes, et je travaille sur les paramètres morphologiques, physiologiques, biochimiques, moléculaires, et ainsi les paramètres de rendement. Mais de faire tout ça dans la même présentation, c'est assez beaucoup. Et merci.
Okay, just for translation, my colleague Mohamed Al-Mukhtar just asked the presenter about the possibility of having results concerning yield of the wheats of the 4 varieties, and the presenter said that it's only a part of the project of the results, and the time is not enough to present all the data, and maybe next opportunities we're going to have those data. Okay, is there any question? Other question?
Une petite question. C'est possible. Je demande seulement l'utilité le flux électrolyte, que vous l'utilisez beaucoup. Je demande l'utilité d'Yellow, son utilité. Et voilà, c'est bon.
Tu peux te présenter juste pour que l'audience puisse savoir qui vous êtes?
Amina Kaldoudi, une doctorante à l'UMF de Fès.
Très bien. Oui, Amina, bienvenue. Oui, les fluides électrolytes, c'est un indicateur des dégâts au niveau de la membrane. La dégradation de la membrane de la cellule qui a permis beaucoup de fluides électrolytes. Les solidité, ils sortent de la cellule. D'accord? C'est un paramètre essentiel de valoriser est-ce qu'il y a une dégradation de la membrane ou non. Chez les variétés, ce stress.
D'accord, merci. Et concernant, monsieur, concernant les études moléculaires que vous faites, vous cherchez les gènes de résistance à la salinité ou quoi? C'est juste une petite une curiosité scientifique, si vous le permettez de me répondre. Sinon,
oui. Oui, les recherches prochaines moléculaires, on cherche les gènes exprimés en cas de stress salin, qui sont les gènes qui s'expriment dans le temps de stress salin. D'accord?
D'accord. Merci.
Okay, thank you very much. Thank you, Saïd, for the presentation and answers also. Now it's time to go back to our colleague Mohamed Abdullawi.
Oui, bonjour, monsieur.
Are you? Bonjour. Do you have the presentation ready? Just okay.
Oui.
Share your presentation. Okay, put it on the full screen. Full screen. Là. Voilà. Allez, vas-y, tu as 10 minutes.
Dear audience, it's my pleasure to present for you my research work on the term impacts of water stress and productivity and quality parameters of weight and their potential use in breeding program. At work, that's part of my PhD in collaboration between the Faculty of Science of Penetra and the National Institute of Agriculture Research, Rabat. This presentation will be centered around 5 axes. First off, I will begin with an introduction and the objective of our study. Then in first axis, I will be getting into material methods and before concluding, I will present the results obtained and discussion which follows. The cereal sector occupies the primordial place in Moroccan agriculture, of which wheat bread is most consumed cereal. Hence, we must give special emphasis to this crop. From the point of view of production, bread wheat is the second most important crop in the world after rice and the first at national level. Given this important bread wheat has attracted particular attention, especially in the breeding programs of Mediterranean countries, including Morocco, because of its adaptation to semi-arid environments. However, bread wheat production is affected by environmental factors through several abiotic stress, mainly water stress, and also attacks by pests and parasites, leading to a drop in yield and the quality, and consequently to considerable and economic loss for farmers. But the main objective of our study was evaluate the impact of drought on the productivity of bread wheat to determine the main selecting criteria for drought tolerance in Moroccan environments, to identify the best promising line resilient to stress and of interested to farmers in Mediterranean regions. The genetics materials consist of 31 varieties and lines to breed wheat from the National Breeding Program of the National Institute of Agricultural Research. The cereal were performed into cereal growing regions with contrasting rainfall on 2 experimental sites. The experimental design is a complete randomized block with 3 replications. Each elementary plot is made up of 6 lines of 5 meter length and 0.2 meter spacing. 5 productivity and quality parameters were analyzed: grain, yield, biomass, number of fertile hairs, 1,000 grain weight, and protein content. The first station is located in the Rabat Salah Netra Mershoush region, known for its humid and relatively favorable climatic conditions, greater than 400 millimeters. The second station, Jamaat Shem, is a part of the arid agro-ecosystem of the Asafi region, with an average annual rainfall of less than 30 millimeters. After maturity, we proceeded to collect the linear method for analysis of different parameters of each site. Statistical analysis were performed using GenStat 18, one-way and two-way analysis of variance, and correlation analysis. The 2020 and 21 agricultural campaign was generally favorable. The national cumulative rainfall reached 271.9 millimeters with good temporal and spatial distribution against 286.9 millimeters for the average of the last 30 years, with a slight decrease of 5%. favorable station Marshoush recorded 367 millimeters, while the arid station Humatsheim accumulated 201 millimeters. The average yield at Marshoush was 57.3 quintals per hectare, while at Humatsheim it was 10.7 quintals per hectare. The genotypes give better yields in Marshoush than in Humatsheim. Indeed, the Marshoush station represents the favorable environment for cereal production. The rainfall usually exceeds 400 millimeters per year, and the other hand, the Humatsheim station is located towards and south in arid area, where the annual rainfall does not exceed 200 millimeters per year. Genotypes evaluated show different performance from station to another, as can be noticed. The genotype of the line BT19I5 and BT19A21 and CCBT 175 and CCBG showed the best results in terms of yield in Humatsheim station, with an average yield of 14.2 quintals per hectare, while BT
you still have 3 minutes.
Okay.
You still have 3 minutes.
On the other hand, his average protein content in Marshoush was 30.60%, while it was 70.09% in Humatsheim genotype. Genotypes give better quality results in Humatsheim than in Marshoush, and the best genotypes in Humatsheim it's CCBT 108 and CCBT 144. The person correlation analysis showed a strong association between yield, biomass, and number of fertile areas, and under dryland conditions. Biomass is an important photosynthetic parameter for the plant, allowing it to generate higher productivity. These two parameters, biomass and number of fertile areas, could be the potential selection criteria for drought tolerance in Mediterranean conditions. On the other hand, a negative correlation of biomass to protein content, this could be related to the absence of rain during and the grain-filling period, which favors competition between the different organs. At Marshoush, of fertile areas under relatively favorable conditions. The results of this study show a significant impact of water stress on waste productivity under Mediterranean and Moroccan conditions in particular. The number of fertile areas and biomass could be potential selection criteria for drought resistance. These trials will be repeated for several years under other stress intensity and agroecosystems, and by integration other parameters to confirm the results obtained. The line BT18I5 showed a high yield potential under both conditions, may be of great interest to farmers, and could therefore be presented to the national catalog for reliance. Thank you.
Thank you very much, Mohammed Abdullawi, and maybe we're going to put your name on this variety, maybe. Thank you very much, and yes, we had too many presentations about cereals and salt, actually. and for our Moroccan colleagues, too much خبز والملحة. I wonder if there is any presentation about legumes or fruits. We need some fruits after all those cereals and salt. Thank you very much. For our audience, how about questions? No question? Okay, we move on to the next presentation by Hamza Boras on the investigation of the effect of groundwater quality on crop production in Tadla perimeter. Hamza, are you with us? Hamza Boras. Hamza Boras, I don't think he is in the list. Hamza Boras is not in the list. Okay, unless he came again, we move to the next presentation by Rawa Akrimi on electromagnetic saline water for potato growth and water relation. Right, this is a legume, a tuber actually. And Rawa, are you here? Rawa?
Yes, I'm here.
Yes, okay, great. Can you share your presentation with us?
It is okay.
I'm waiting for the picture actually, and yes, now activate the modification and put it on the full screen.
It is okay now. It is okay.
Not yet. Activate the modification on your file. Not to have problems after all.
Okay. Okay. No.
Okay.
Okay.
Reshare again. It's not shared. Please share again.
Okay. Okay. No. It is okay. It is.
Full screen.
It is okay now.
There's a delay for.
It is okay now.
Okay.
Okay.
Go.
Okay.
First 10 minutes.
Okay. Firstly, I would like to thank the organizers for giving me the opportunity to participate in this event. Here I will present my work entitled Electromagnetic Saline Water for Potato Growth and Water Relations. The presentation includes a background of work, details of methodology, results, and conclusion. Want to choose? We know that salinity caused by saline irrigation is one of the major constraints limiting plant growth and productivity all over the world. In the world, there is 1/3 of cultivated land affected by soil salinization. In the case of Tunisia, there is about 50% of irrigated areas are at risk of salinization due to use of saline water. These areas cover around 1.5 million hectares. It's to say 10% of the country's area. Salt-affected soils are mainly located in the center and in the south of Tunisia, given the aridity of climate, the high temperatures, low rainfall, facing the current climate change. The main agricultural sector challenge now is the allocation to saline water to successfully grow crops. Those Soil salinization caused by saline water irrigation adapt to severe climatic conditions, and the growing demand for water make the need of more efficient and less expensive desalination methods are very required. In this context, we studied the effect of the physical treatment technique, which is the electromagnetic treatment of saline water, and potato growth and water relations. The general aim of this work is to assess whether electromagnetic saline water can alleviate salinity impacts in potatoes, and to evaluate the influence of electromagnetic saline water on plant or water relation, growth, and yield, and also to verify the applicability of this technique in case of dry conditions of Tunisian spring season. The study was conducted at the center of Tunisia, especially in the Regional Center of Agricultural Research of Sidi Bouzid, with the planting date at the 1st of March and the harvest date at the 4th April. The experimental area had semi-arid climate with 2,000 millimeter precipitation and average evapotranspiration of 1,200. The experimental layout had a factorial randomized complete block design with 3 replications and 2 factors with 3 levels each one. The first factor consists of 3 potato varieties, Punta, Bellini, and Alaska, and the second factor includes water treatments with groundwater as control treatment and saline water with 8.5 electrical conductivity and saline water with the same electrical conductivity that will submit an electromagnetic treatment. The electromagnetic treatment was done with for the 60A series device, which is composed of an electromagnetic tube designed to transmit electromagnetic signals onto the circulating water and connected to an electromagnetic box pre-programmed to generate electromagnetic signals. The measures done in this study includes agronomical traits, which are plant height, plant dry white, and fresh white, and the yield per plant. The traits to measure to evaluate the plant-water relations were leaf relative water content, leaf water potential, water photosynthetic water use efficiency. We also quantified the proline concentration in leaf tissues. The results of this study revealed that the plant height and the dry white per plant were increased with electromagnetic saline water as compared to non-treated saline water in Spunta and Alaska varieties, but there is no significant difference between the 2 saline treatments in Bellini variety for plant height and dry white. The fresh white of Bellini variety was covered with the electromagnetic saline water. The evaluation of yield per plant revealed that fortunately, the 3 studied varieties had a significant improvement of tuber yield with electromagnetic saline water, while conserving the highest yield for the control treatment. Measures of relative water content also revealed an enhancement of the relative water content with electromagnetic saline water compared to non-treated saline water for the 3 assessed varieties, whereas the water potential and the photosynthetic water use efficiency were enhanced only in Spunta and Alaska. Measurement of proline content in leaf tissues revealed that the concentration were in the decreasing trend of saline water, electromagnetic saline water, and finally groundwater for Alaska variety, but there is no significant difference between the 2 saline treatment for Spunta and Bellini. From results of the study, the results of the study demonstrates that electromagnetic water improved the relative water content and water potential for Sponta and Alaska. These two attributes were linked to plant height and improvement on plant height and fresh and dry biomass accumulation. The yield of the three studied varieties was also enhanced by electromagnetic saline water compared to non-treated saline water. Even though further research are required to ensure safe use of electromagnetic water and to identify the exact mechanisms governing the plant response. Thank you. Thank you.
Thank you very much.
Okay.
Thank you very much. We still have 3 minutes to the discussion, but we're going to use those 3 minutes, additional 3 minutes into the discussion. And I ask the audience if is there any question to our colleague Rawa. Our colleague Rawa, I think you are from Tunisia. Is that okay?
Yeah.
Welcome.
Thank you. Thank you.
Anyone interested in salted potato?
Yes, dear Aziz, dear Khalid, yes. Okay, I have just one question regarding the technology, electromagnetic water. Is it accessible for farmers, or is it expensive to use in the field?
The first question, you put planting in March 1 and the harvest in April 4. I cannot imagine potato for only 1 month. It's not possible. Potato need 3-4 months. It depends about season and the variety. Now, can you explain this first aspect? Planting March 1, harvest April 4. And I have the second question. The second, the important parameter for potato is the caliber, the commercial caliber, not the yield and the other parameter. The important for the farmer, the commercial caliber, and there are nothing about the caliber that you will go to have. You can have a good and increase of yield, but not the commercial caliber according to and thank you.
Okay, just for the organization issues, I think I'm just a little bit confused. We have the first question from Mohammed Ait Mukhtar and 2 questions from our participants in the yes. Okay.
Yeah.
Okay. So we have another questions. Okay, go ahead. From our participants, Professor Pilar Morera. Okay. Can you answer the first one for Mohammed Ait Mukhtar question?
For the date of planting, I did mistake. There is the planting in March and the harvest occurred on the 4th of June. There is the 4th month of the growing period. Concerning the second question, which is the caliber of tubers, yes, I did this measure and the commercial and the marketable ones and the proportion of non-marketable and non-marketable tubers, but I didn't present them here.
Okay. I think those answers are enough for our participants. Any other question? Thank you, Rawa.
Okay, thank you.
And now we're going to the last but not the least presentation by our colleague Nassima Darhal on the productivity and nutrient status of forage corn and fodder beets irrigated with saline water. Again, cereals and fodder beets. Nassima, are you with us? Nassima? don't know if she is in the list or in presential.
I don't think so. In the list,
she's not in the list.
Yes.
Nasima? Nasima? Okay, so actually I see that we have two missing presentation actually from Hamza Boras and Nasima Darhal. And if we don't have any sign from them, we're going to close this this panel. And just, Si Mohammed, do you have any concluding remark about this panel? Anything you can add?
As you said, it was a lot of cereals, but it's okay for today. Maybe we could have other fruits, vegetables, and the other sessions. So yes, there are two missing people. So I checked in the list down here. I think a lot of study in this session were about the effect of salinity on crops, different crops, physiological, biochemical, particularly, and with trials in the field and others under controlled condition, and that the most studies related to salinity followed the kinetics observed in the majority of the study in this axe. One study was related to water stress, and it was also interesting to select the varieties which are more tolerant to water stress to use in different parts in Morocco. I think the presentations in this session were very, very important and provide us with a lot of insights to help the researchers in their topics of research. Thank you.
Thank you very much, Professor Ait Al-Mukhtar, about the concluding remarks. Just to add some elements, just to say that most of the presenters are PhD students, postdocs sometimes, and I think that they are not presenting all the whole work. It's a process, and also there is a lot of work, and all the data is presented today. And it's a process, as I said, and maybe in next future we will have more data about the reaction or the response of the crops or the soil to the soil salinity or water salinity. We still focus on cereals. We still focus on biochemical parameters. And as I saw, I can observe that some data on the economical cost, economical analysis are missing. Sociological impact of the salinity is also missing, and I think that those traits could be dealt in other sections, and maybe in other panels, technical panels, we will have those kind of details on the studies. Thank you very much for your presence and for your patience also. There were a little bit 6 presentation. We are missing 2, as I said before. 6 presentation, and I think that we learned quite a lot about the impact of salinity on cereals mainly, and I hope that we will meet together again in physical, not in hybrid. And I hope that next time I will come to Laayoune and be hosted by Moroccan hospitality. As we know, there is warm hospitality in Laayoune, I guess. Thank you very much, and see you then. See you in the see you tomorrow. Yes, see you tomorrow. We still have tomorrow program. Thank you very much, and have a nice evening.
Thank you.
Okay, yeah. Okay, it's okay, yeah. Okay, ladies and gentlemen, thank you to reach this final session for today. Now we have to start our session. The next presentation will be our next presentation. Normally. Okay. I think next presentation will be the presentation of Zainab Ktiri, with salt. Zainab Ktiri, do you hear me? Are you here? She is connected? No? Not connected? Yeah, we can go to the next one. Yeah, I think the next one it will be Iman Nabulsi. Dr. Iman Nabulsi, are you connected?
I think she's in another room. Maybe she may be coming.
Huh?
She may be coming.
Yeah, it's our.
Maybe another Iman also. Okay.
Yeah, it's now. It's our. For the session A. Yeah. Iman Nabulsi? No. She's not connected? So, to go to 17. But I don't know if we are at time or not. On time?
Maybe.
Yeah, Caesar currently.
They're still waiting for the copy.
Yeah. But the time is.
I can go and look for it. Okay.
Yeah. Iman Nabulsi, she's not here. The other one is not here. And this it. Yeah. Kamel Nakaz. The next speaker, Kamel Nakaz. Huh?
Not Kamel Nakaz, it's Ines Toumi.
Who?
It's Ines Toumi. Nakaz Kamel is a co-author.
In my plan, no, in my plan there is Iman Nabulsi, then we have Kamel Nakaz.
Not Kamel, it's the presentation for Toumi Ines, and Kamel is a co-author.
Yeah, who will present?
Toumi Ines.
Toumi Ines.
Yeah.
Are you ready?
Yeah. I will show you
you can share your presentation.
Yeah, it's okay.
So our next speaker will talk about response of early maturing peach Prunus persica grown in arid area under saline water to partial root zone drying. Yeah, Ines Toumi.
I will share my screen.
Yeah.
Sorry.
Okay.
This is me.
Dr. Ines, your screen is not yet shared. Not yet. Please try to share your screen.
Yes, I will share.
Okay.
I have a problem in the connection.
No. Yeah, we will let you solve your problem, then we will return back to you after the next presentation.
Yes, it's okay.
Yeah,
It's okay.
concentrate, yeah. Then the next presentation it will be for by Kenya Donkato. Kenya Donkato.
Hello.
Hello. Are you ready?
Yes, I will share my presentation.
Yeah, please share your screen.
Can you see it?
Not yet. It will be, the presentation it will be on evaluation of the nutritional status and the food potential of Salicornia bigelovi grown in the United Arab Emirates.
Can you all see my screen?
Yeah. Okay. We lost it. Yes.
Now?
Yes. Yes.
Can you see my screen?
Yes, please put it in the first screen. The presentation
How
mode.
do I do that?
Not in the bottom. In the bottom. The bottom of your screen, you have a symbol of screen.
No.
Yeah, that's okay. Go ahead.
Okay,
The
thank
floor
you
is
all.
yours. Okay.
First of all, can you see my screen now?
Yeah, go ahead.
So, first of all, let me thank you all for being here today. My name is Kenya, and I'm going to present the study entitled Evaluation of the Nutritional Status and Food Potential of Salicornia bigelovi grown in the United Arab Emirates under the supervision of Dionisia Lira and in collaboration with Maize and George. Over 30% of groundwater resources are saline and MENA region, and 70% of groundwater is saline in the Gulf countries. So there is a need to adopt Biosaline agriculture to improve land productivity. Halophytes are well adapted to salinity and can improve food. I'm sorry, I cannot see my whole food availability in marginal areas. I will do, sorry. Salicornia bigelovi is one of the most popular commercial halophytic crops, commonly sold fresh in North America and European markets. These pictures here, they show the production of Salicornia bigelovi in United Arab Emirates. The present study aimed to evaluate the nutritional composition of 3 vegetative stages of Salicornia bigelovi, and seeds as well, and select the most appropriate for human consumption. It's not passing, sorry. I don't know what's up to my
We are hearing.
my presentation. No, the problem is that it is not passing the slides.
You mean you are not able to go forward?
Yeah, you can't progress?
No, I cannot. It just stop it here. It's just loading in my computer. I don't know what I do. I will stop and...
You can start and share your presentation, then reshare it. Yeah. Try again.
So for the methodology, we cultivated the plants in the experimental station of the International Center for Biosaline and Agriculture, ICBA, as you can see in the picture, in the United Arab Emirates. So we cultivated 11 genotypes of Salicornia in field plots, and the plants were irrigated with saline groundwater, an average. So we grow along all the life cycle of Salicornia, and we took only the fresh tips between 10 and 15 centimeters, and we collected in the early vegetative stage, the middle vegetative stage, and the late vegetative stage, as well as the seed stage. Vegetative shots and seeds were analyzed for their nutrient content composition. By that I mean macro and micronutrients, amino acids, and fat acids by food allergens laboratory in Cyprus. The three vegetative stages were compared by ANOVA as well as compared to seeds. The listed parameters there were transformed to fulfill the assumptions of ANOVA. And when we found significant differences at ANOVA, we performed the Tuckey test, considering a 5% as level of significance for all the statistical analysis. To evaluate how nutritious Salicornia was for different stages of life, we select set age groups to calculate the percentage of nutritive contribution that 100 grams portion of Salicornia would provide. So we calculate the percentage of nutrient composition as the average value of the measured component in Salicornia, and we divide it by the daily recommended intake, which is a standard number, and we multiply by 100. So this is standard number, which is the daily recommended intake. It will depend on the age group, and it's according to the Institute of Medicine guidelines. Comparing vegetative stages with seeds. So, as it's presented in this table, the lowercase the comparison between the vegetative stages, and the capital letters are the differences between vegetative stages and the seeds. So it's highlighted here, energy, carbohydrates, and total fiber, because these parameters, they tend to increase as the plant grows. And calorie content is the mainly from the increased carbohydrate starch content. This makes it also more versatile for food processing in manufacturing. It's also important to point out that most of the parameters in the seeds were higher than in the vegetative stages, particularly the protein. It stopped again. Sorry, it's not working again. Oh no, now it's working. So in this table, I presented the macro micronutrients, ashes, and vitamins. And as you can see, the ash increases as the plant matures. And this is an indicative of high nutrient composition, mainly from sodium. And it's pointed out here that sodium content of this alophyte is one of concern, and it's too high when compared to other vegetables. But the values that we found here, they are inside of the range that's okay for consumption for humans, except the mature vegetative stage. Regarding to the amino acids in Salicornia, as you can see, Salicornia, it provides all the essential amino acids to consumers, and it's pointed out here with the green arrow that lysine and methionine, which are normally limiting in plant-based crops, are presented for Saliconia bigelovae. So many legumes and vegetables are usually low or deficient in methionine, so Saliconia would be a good alternative. And this plant complements grain-based diets as it supplies good levels of lysine. Fat acids. So we had a very huge list of fat acids, but here I only presented the most prominent ones. For example, linoleic acid.
You still have 3 minutes.
What? Are you listening to me? Are you seeing my screen?
Yeah, you still have 3 minutes.
I'm sorry. Okay. These are presented, and we also calculated unsaturated ratio, and between the range that we found between 3 and 4, it's high compared to other oils, and it's edible since it has nut-like taste and texture similar to olive oil. And related to oleic and linoleic ratio, it regards to the shelf life of the product, and it's a little bit low. Compared with other vegetables, Saliconia has more fiber, sodium, ash, calcium, magnesium, and vitamin C than certified data from spinach and asparagus. And the impact of consumption on our age group, Saliconia, as you can see, we calculate the daily recommended intake, and the first batch of vegetative stage in the seeds, they provide from partially to almost fully the needs of a child between 4 and 8 years old. And I would like to highlight the vitamin C, which supplies 93% of the recommended daily intake. And vitamin C is an immune-boosting property. It has anti-inflammatory, antioxidant properties. It's important in collagen synthesis and growth, and also for wound healing. So this halophyte is very important for people living in places like roof areas, because people usually they don't eat as much vegetables as they expected. So in conclusion, Saliconia provide highly nutritious food source along its life cycle, especially the early stages. The vegetative shots have more fiber, sodium, ash, calcium, magnesium, and vitamin C than asparagus and spinach. And the early vegetative stages and the seeds of Saliconia can supply partially to totally the daily recommended intakes for children living in marginal areas. And the potential growth of Saliconia using saline water, strengthening the food security income in coastal areas. I would like to thank you all very much for your attention.
Thank you much for you too, and thank you much to be on time, Kenya. So, if there are some questions for our speaker.
Yeah, I have a question.
Yeah. You can, the micro is here, One is, there is a question for you.
Yeah, Kenya, good information about Salicornia. This is good plant. Just maybe if you have idea how much water is requires to grow Salicornia. Number one question. Number two is, what are the constraints for adoption to plant in larger scale? Because when I see your data, it has very good amount of sodium uptake, which is very good for the saline condition. If it requires less or somehow reasonable amount of water, then that makes life easy for its adoption. So if you have idea, please elaborate those two things.
Thank you. The first question was about how much saline water Saliconia requires. Unfortunately, I don't have this information. I was more concerned about the food status of the plant. But I believe that, for example, I worked with Salicornia nay in Brazil, and it doesn't require that much water. But when you grow Salicornia in more arid areas, you need more water to release the accumulation of salts in the top of the soil. But I don't know the exact amount. I'm sorry that I can give you this information. But my supervisor, Dionisia Lira, she has many works with Salicornia in arid areas. So probably you can find this information in one of her papers.
Well,
Did I answer your question?
the second question, what about the constraints for upscaling? It's not. Yeah.
Could you clarify that, because I didn't understand the question?
Yeah, if there are some constraints that that limit the adaptation of Salicornia or upscaling.
That's very interesting. No one ever asked me that before, but it will depend in
For extension.
which way are you...
For extension, cropping extension.
Yeah, no, but I believe it will be very interesting to grow Salicornia not only in soil, but also in water. And it grows faster in water than in the soil. So I think it would be a good option to upscale the production of halophytes. But regarding to like field crops, I honestly I don't know like the
Thank you.
springs.
I can add
Yeah, thank you very much for your
Sorry,
presentation.
I can add some information about the water requirements. I can.
Yeah.
Salicornia, Salicornia is a hydrohalophyte. It means that need high amount of water. If you look at the nature, also you can see the Salicornia near the saline water. After that, you can see another crops, another halophytes. Then this crop needs more than 3 meters a year water during the growing cycle. Okay. I think the main obstacle for upscaling of the crop is the high water requirement. It means that the Salicornia need high facility for irrigation because maybe you need you should irrigate it twice a week. It means that you cannot upscale in a large scale. I think the main obstacle for extension of the quinoa high water requirement and this make a problem for irrigation. Thank you.
Thank you very much.
you.
Yeah, of course. Yeah, it's a halophyte. Yeah, it requires more water. Yeah. Thank you.
So that would be an interesting thing to use it in integrated systems.
Yeah. Could you repeat what you said?
No, I just said that since Salicornia requires a high amount of water, it would be interesting to grow it in integrated systems like from fish or aquaculture facilities.
Thank you for this additional information. Yeah. So, thank you, Kenya, for your presentation, for your answers. Now we have to move to the previous presentation that we lost with Ines Toumi.
Yeah.
Yeah, are you ready?
Hello.
Yeah. Please share your screen. And go ahead.
Yeah. Hello, everyone. Thank you for giving me this opportunity.
Your screen still not shared, please.
But it is shared for me.
Yeah. Yeah, there's a problem. We try to solve it. Okay.
Share it for me.
Yeah, okay.
It's okay?
Okay.
Yeah.
Go ahead.
Hello, everyone. Thank you for giving me this opportunity.
Please, could you put it on the full screen? Diaporama.
Yeah.
Not yet.
It's okay?
No, not yet. Try. Mode diaporama.
Yeah, it's in this mode diaporama.
No, it's not. Yeah, go ahead. Go ahead, no problem. Okay.
Okay. I'm Toumi Ines from Arid Regions Institute, Tunisia. My presentation entitled Response of Early Maturing Fish Grown in Arid Area Under Saline Water to Partial Erosion Drying. As you know, water is the main limiting factor for crop production in dry area. Current and future climate change induce a decrease in water availability and an increase in temperature. With increasing water scarcity, irrigated agriculture needs to be more efficient in using water. Deficit irrigation strategies based on water supply restriction were proposed as promising techniques for water saving. Fish is...
Please, your slides are not moving. Are you still on the first slide?
For the introduction.
Yeah, no, the first slide. We are in the first slide with the title. Please, you can share your presentation, then share it again.
It's okay or not?
No, we are still on the first. We are still in the first slide. No, it's not presentation. Yeah. You can change the mode from mode presentation mode or full screen.
Wait.
Okay.
It's okay?
It's okay. No.
Yeah. I will begin for the introduction. Donc, water is the main limiting factor for crop production in dry area. Current and future climate change induce a decrease in water availability and increase in temperature. With increasing water scarcity, irrigated agriculture need to be more efficient in using water. Deficit irrigation strategies based on water supply restriction were proposed as promising techniques in water saving. Fish is the fourth most important fruit crop in the world. It represents the main deciduous fruit species cultivated all over the country. Fish cultivars are characterized by an important need of water irrigated. Early maturing fish have grown in recent years and constitute one of the biggest consumers of irrigation water. In this context, our work search is aimed to evaluate the use of partial erosion drying and their response of the low-chiming cultivars to the irrigation strategies. Donc, I will pass to the methodology. The experiment was carried out in a commercial farm situated in southeast of Tunisia, especially in Tataouine Governorate. The climate is an arid climate with hot, dry summers and excessively low rainfall. the cultivar used is Flordastar grafted on Garnier rootstock. The row spacing is 25 meters square. Trees were goblet trained. The water salinity is around 3.17 millisieverts centimeter, and the soil is sandy loam with very low organic matter. Two irrigation treatment were applied. The full irrigation treatment when trees were irrigated at the full cultural evapotranspiration, and the partial zone drying consisting in alternating irrigating one side of the tree at 50% of the evapotranspiration culture every 10 days. The trees were drip irrigated with 2 lateral lines per tree row using 4 meters per plant. This study was conducted 3 consecutive growing years. For the main results obtained in this study is about fruit size, fruit weight, fruit yield, and water productivity. The fruit diameter and the fruit weight is highly enhanced in the full irrigation treatment during the 3 consecutive years. Water productivity was enhanced in the deficit irrigation strategies, mentioned the partial root zone drying. For the fruit quality traits, dry matter content, soluble solids content, and total phenolic contents was determined in this study. For the soluble, for the dry matter content, this parameter was significantly enhanced by deficit irrigation treatment expressed by partial root zone drying irrigation strategies. But for the soluble solids content expressed by degree bricks, the same results for dry matter content. The partial root zone, the fruits irrigated with the partial root zone drying where the fruits with a high level of SSC. Finally, for the third parameter determined in this study, for the total phenolic content, the phenolic content is a secondary metabolite for the peach fruits. This parameter was enhanced by either deficit irrigation treatment expressed in the partial root zone drying compared to FE fruits. For conclusion, the application of partial root zone drying during the growing season for annually maturing peach cultivar seemed to me a promising tool for irrigation water management in an arid and warm area. PRD irrigation strategies lead to a significant irrigation, water saving, and increasing water productivity. Also, it improves some quality products parameter and thereby the farmer's economic return. And finally, thank you for your attention.
Thank you for you also too, for your presentation. We'll open for if there are some questions. Please online if there is someone who has a question. The floor is yours. You can go ahead and ask your question. I think there is one question. Yeah, you have it. Yeah. There is one question for you.
Good. It is very practical experiment. My question to you is, you have significantly less yield under your partial irrigation system than full irrigation system. If you see your yield data there. Yes? Hello?
Yeah.
But you have significantly good, higher water productivity, but in terms of yield, you have significant yield penalty with your partial root drying method. And in your conclusion, you said partial root drying method is good. Yes, you increase the water productivity, but your yield penalty is very high. How you can make this conclusion? Because if always farmers, they care more for yield and profit. How you will justify that?
Yeah, the water productivity is clear, enhanced by the partial root zone drying. The using of Laayoune variety such as Florida Star Cultivar mentioned in this study was this is a very earliest cultivar. The economic value of the fruits is highly compared to the other fish fruits for the early late or early season cultivars. For this we accept this reduction in the yield and in the fruit size.
Yeah.
My question was, maybe you go in your slide where you have presented the grain yield. Go above.
Yeah.
Go above.
More.
Yeah, in this thing.
Yeah, when you have the fruit.
Yield, yeah, that table, you are right. Click again that table.
The table. Yeah, this one.
For the table.
Yeah.
For the table one.
Yeah.
Yes, see. In all 3 years, you have significantly less yield, your fruit yield, under partial root drying method. See, in 2013, you have a difference of almost 7 kg. By the way, you guys need to present those data in standard unit, not kg per tree, kg per hectare or ton per hectare, okay? When you write paper.
Yeah.
And 2014, see almost 10 kg and 15, 4 kg, even 4 kg is already significant. 10 kg is huge difference. Then farmer will be in loss if you recommend your partial root drying method.
Yeah.
You understand?
No, not clear. Not clear the comment. Please repeat, sorry.
What's the question?
Okay, you see the yield. In 3 years, all 3 years, you have significantly less yield under partial root drying method. You increase the water productivity because you applied less water in partial root drying. But as you have huge yield penalty under partial root drying, farmers will not agree with your finding. Okay? Other people will not agree with your finding. My question is how you will justify this? And you said that in your conclusion, partial root drying method is promising. If I see this result, I will not say I agree with your conclusion. This is not promising.
Yeah. You saved water. With this technique, you saved water, but also you have a decrease in yield.
Huge decrease.
Yeah. huge decrease.
Yeah.
But
Yeah, the decrease in the yield was composed by the fruit quality in the first step, and for this cultivar, Flor-d'Astar cultivar is a very early maturing peach cultivar. Donc their commercial value is very high compared to the other fruit harvested from the late season or the late cultivars.
thank you very much. Yeah, thank you for your presentation.
For this reason, we accept this reduction in the fruit yield.
Yeah, thank you. Yeah.
Thank you.
Yeah, it's economically valid. This. Yeah, because the decrease is huge versus the saved water. Yeah, I don't know if the farmers will accept that or not. Anyway, thank you very much. Thank you for your answer, for your presentation.
Thank you.
Thank you. I think it's the latest one, the latest presentation. There are some. We will try for some previews that were not present. For Zainab Ktiri. Zainab Ktiri, are you here? Are you connected, Zainab Ktiri? Zainab Ktiri, that we missed. We can go to Iman Nabulsi. Iman Nabulsi. So we have to, we have to close the session. Thank you very much for attendees. Thank you to attend this session. We have to, it's the latest presentation, so we have to close this session. Thank you very much for your participation. Thank you.
Otherwise, we can go home.